how animals can learn complicated things very quickly. And we're, of course, all familiar with learning complicated things very slowly, like trigonometric equations. They take endless repetitions before they sink in. Sometimes they don't sink at all, as in my case, I have to look them up every time. Other cases of complicated things taking a long time are a tennis serve. A professional tennis player probably serves several hundred thousand times before they play their first tournament. And so all these complex tasks, in this case, require a long time, many trials, many experiences of reward or failure. On the other hand, we're well familiar with other things that we can learn very quickly. When a child learns to ride a bicycle, this takes an afternoon. The parent holds the bicycle the first few times, then the child crashes 3 times, and then she figures out how to balance it, and off she goes, and she will never crash in that same way again, right? So it takes about 3 or 4 or 5 experiences of failure before the successful task is implemented. Similarly, language learning in children, when you show the child a picture book with the words, that's a kangaroo, that's a banana, and you do that 10 times, and the child will start saying kangaroo when you turn to the page of kangaroo. So it doesn't require endless repetition for something abstract, like a new vocabulary word to form and get used routinely. So my claim is that in neuroscience, we have focused excessively on these slow tasks that take a very long time to learn. I wrote a review a little while ago to document this. And what I'm plotting here is for different learning tasks that we know about, the amount that gets learned in the course of performing this correctly against the number of reinforced trials that are necessary to learn that. And you can see that there's sort of a cloud of points over here where it takes 10,000 or 100,000 trials to perform the task correctly. And then there's another cloud of points over here where it takes only about 1 to 10 trials to perform the task correctly. And I want to illustrate what these are. So for example, fear conditioning in rodents works very quickly. It's kind of a 1-bit task. The rodent has to associate one stimulus with danger and the other stimulus with safety. And they can learn that after 1 foot shock and retain the memory for hours and days. So this is a 1-bit task. That's the amount learned, and it takes 1 trial to learn it. So I put that point at the 1, 1 coordinate here. Here's the child learning a new vocabulary word. At that scale of that vocabulary, the new word might be 5 bits of information. And it takes maybe 5 trials to learn it. So again, it lies on that curve. Here's a golfer trying to perfect the golf stroke. Again, something that takes professionals many 100,000 trials or so to get close to the green. And you can estimate how much information is involved in striking the ball just so. And so that data point lies over here. There's a lot being learned on the order of 10 to 20 bits, but it takes a long time to learn it. Here's a typical monkey performing an inside-out reach task with pushing a lever in 8 possible directions. It takes many months, sometimes over a year, to train a monkey to do these kinds of tasks correctly. They are really simple tasks. They have to associate maybe 8 stimuli with 8 directions. And so low bit rate, but it takes maybe 10,000 trials to learn such a thing. Here's the task that wins in terms of being at the bottom right of this graph. This is a task that's actually quite popular in mouse neuroscience. You're training an animal to detect the position of an object by moving 1 whisker, and sometimes the object is near and sometimes it is far, and the animal has to lick on the left or on the right, depending on whether the object is near or far. Very abstract task, something that never comes up in the natural life of a rodent, right? And correspondingly, it takes 10,000 trials or so for the performance to saturate, and it saturates at a really low level of somewhere between 0.1 and 0.2 bits. So these animals make a lot of mistakes, even at saturated performance. Here's another task, very popular. There's an entire industry associated with that task, the International Brain Laboratory, where a mouse has to turn the steering wheel to the left or to the right, depending on whether the stimulus looks this way or that way. Again, the performance never gets very good. It's a two alternative task, and they get about 80% correct when saturated. And so that's a bit rate somewhere below 1, and it takes on the order of 10,000 trials or so to learn that. All right, so my point is that neuroscience has spent a lot of effort, including, like I said, an entire factory of people at the Allen Institute and at the International Brain Laboratory, working on the bottom right end of this graph. And we thought some years ago that it would be good to flesh out the top left end of this graph because it's probably more relevant to our life and to the lives of most animals to be able to learn something quickly after a few experiences, not after 10,000. Okay, so the experimental paradigm we settled on was a classic tool, the labyrinth, and we allow a mouse to explore a labyrinth. And the important thing is that the mouse can do this completely unconstrained. There's no trial structure. We're not interfering with it. We're just putting the mouse in a cage. The cage is connected via a tunnel to this labyrinth that has a structure like a binary maze. There's T-junctions at 6 different levels, ending in 64 possible endpoints of this tree. And one of the endpoints has a water port associated with it. The animal is water deprived, so it feels very happy when it discovers that water port. But I should say, it doesn't know ahead of time that there is a water port. It knows nothing. I'm only going to tell you about the exploratory behavior during the first day of the animal's experience with the labyrinth. Okay, so we drop the animal into the cage. We turn all the lights out. This is important. This happens in complete darkness. The animals can't see anything. And then the experimenter walks away. And meanwhile, there's a video camera keeping track of what happens inside the labyrinth. And then we use machine learning tools like Deep Lab Cut to interpret the video and track the trajectories of the mouse. So I'm going to give you a few slides to show you just roughly what the behavior looks like of the animals. So here's an example of a mouse exploring. It still has not discovered that there exists a water port. It doesn't know anything about it. The water port is this construction on the right-hand side over here. Now in a little while, it discovered that there's a drop of water. You saw the red dot. It comes back, but there is no more drop of water because we turned the water port off for about 90 seconds, so it has to go somewhere else. Okay, it continues exploring the labyrinth. And I want to show you briefly what happens next because it's kind of interesting. You'll see that the mouse returns directly from this location to home without making any mistakes. So there's this interesting homing behavior that we found, and I won't talk about it further today, but just mention it that there is this, the animals seem to be carrying around the memory of the route to the exit while they're exploring the labyrinth, even, like I said, the first time they enter. All right, now let's look at the next time the animal enters the labyrinth. So that number 19 is going to go to 20 because that's the 20th time that it walked into the labyrinth. And so it somehow found its way to the water port without making any mistakes and collected the drop of water. Then it continues exploring some more. All right, so to give a kind of an overview of what the typical lifetime of the animal in the labyrinth looks like on the first day. In the beginning, there's a lot of exploration. The animals try to cover the entire space. Eventually, during that time, they discover the water port here. Sometime later, you find that they combine an exploitation run where they go to the water port with some more exploration. Sometime later in the day, they will also just run in from the cage straight to the water port and back out just to collect a drop of water. But really, throughout the night, there is this exploratory behavior where the animals continue to look in other parts of the labyrinth. My guess is that they are expecting that something interesting will happen. They don't know that nothing will happen anymore, but their expectation, their baseline probably is that the world is a changing place and it's worth exploring.
So here, they've been in the maze for 1 hour.
Correct, yeah.
Continuously.
That's 53 minutes of time in maze. Yeah, I'm not counting the time in the cage. They keep going back and forth between cage and maze.
But actively, it's not that you put them back.
No. Completely under its own. The animals like exploring. They spend more time in the maze than in the cage. Interestingly, even if you put the water into the cage so they can drink whenever they want and they know where it is, they still go into the maze about half the time. I think they're optimists. They think that this mysterious environment has to hold some resource that I don't know about, and so they keep going back to look. All right. I showed you an example of a mouse that took a long time to discover the water port, but once it discovered it, it ran back there reliably the next time. And this is not an exception, so here are just examples from a few other mice. I'm plotting the duration of the first water run, which means the time in seconds from entering the maze to finding the water port. Yeah? And then the next data point is the second time that happens. Yeah? So you see the first time finding the water port in this case took 900 seconds. The second time took only a few seconds. This is when the animals almost always can do a direct run to the reward location. And again, here's another example. Long time to first discovery, much shorter to second discovery, and then very quickly the animals settle on the most efficient route to the water port. Another example here. Okay, so at this stage, I'm guessing that half of you are thinking that obviously the animals are using olfaction. We know that mice have a very good sense of smell, and maybe what they're doing is when they find the water, they pee on the floor, and then they just go back to where that drop of pee is. So that it's a little bit like the Hansel and Gretel story. I don't know, is that familiar in China? But where the children drop breadcrumbs along the trail in the forest in order to find the trail again on the way out. Yeah. So maybe the animals are doing something like that. And this occupied us early on, and I'll show you one experiment that kind of puts that to rest. You can see that the symmetry of the maze is such that you can take the maze and rotate it 180 degrees, and you just have to open one door and close another door, and it's the same structure. Yeah? And so let's suppose that the animals early on deposited some odor cues here near the water port. When you rotate everything, they should now be led to this location here, right, which was the symmetric location to the water port. Yeah. So does that happen? Let's just look at an example of one animal that's, this is towards the end of the night, so it's gotten quite expert. It does these trajectories to the water port, a little bit of exploration back out. Here are a couple of trajectories on subsequent runs where it goes directly to the water port and back out. So then the mischievous graduate student took the maze and rotated it while the animal was waiting in the cage. And then the question is, what happens next? And on the next trajectory, the animal again goes to the correct location of the water port, not the location to which it would have been led if it had been following odor cues. We also did more comprehensive experiments where we ablated the animal's olfactory receptors. This isn't very hard to do. You can inject something in the nose that kills, temporarily at least, all of the olfactory receptors. You can prove the animals can't smell anything, and they learn at the same rate with the same one-shot learning performance as the normal animals. Okay, so it's not smell, it's not olfaction. Yeah. So what is it? What is the mechanism by which they can repeat this trajectory to the water port perfectly without actually having ever executed that exact trajectory? Because the first time they get to the water port is a very complicated route with many errors, goes through the labyrinth. And then on the next trial, they somehow are able to cut out all the complications. So how is that possible? Okay, so there's a hint here that you can get from comparing the experience of different animals. So what I've plotted here for about 10 different mice is a timeline of water rewards that they collect. So every dot here is arrival at the water port and collecting a reward. And so you can see that this animal took a long time to find the water port, but then the next time this only took a few seconds. Yeah. On the other hand, here's an animal that got really lucky. It discovered the water port by accident very early on, but then took a long time to be able to use that knowledge and come back to the water port and then start to use it routinely. Here's another example of an animal that got lucky, but it still had to wait some time before it could use the water port at a high rate. And so the suggestion was that maybe the animals learn something already before they ever discover that there's a water port. Maybe they need to learn something about the environment that then allows them to make use of this solution that they found suddenly and reuse it. And so one prediction is that if you allow the animals to learn about the environment without a water port present, so all they can do is walk into the labyrinth and walk around, but there's no reward location, there's no special place, and then allow them to find the water port, they should be doing better. They should actually be able to make use of that knowledge that they acquired about the environment. So what we did is on day one, we allowed the animals to explore a labyrinth that was empty, no water port. Then on day two, we put it into the conventional labyrinth, and they started collecting water rewards right away. So if you look at the timeline here, it takes a naive animal about an hour or so, maybe 3,500 seconds, to get to the point where it can routinely use the shortest route to the water port, whereas the animals that are experienced the day before, just learning the environment, not learning the solution, not learning the particular location of the reward, can start to exploit that right away.
Something I don't understand is that the initial points, the statistics of the first time they encounter should be similar, no? I mean, maybe not because they don't explore the same way.
Yeah, exactly.
But they don't explore the same way, but still I don't see why it's shifted so much to the
Yeah, okay. All right, thanks for that question. So the question is why? Why is that? Why do the naive animals take so much longer to even discover it and then be able to use it? And so it turns out to be because they explore the environment very inefficiently. So here's a naive animal entering the environment, and I have to explain this raster graph a little bit. So the horizontal axis is time, and the vertical axis is the node in the labyrinth that the animals arrive at, where by node I just mean the end nodes. There are 64 locations at the ends of the tree, and I'm marking which of those locations the animal is at. And for convenience, I've colored the northwest part of the labyrinth in red and the northeast in blue and so on. And so we can see now how the animal explores the space. And what you find is that the naive animal starts out exploring very locally. It's in some sub-octant or subspace of this red area. Then it moves to the other half of the red area. Then it moves to the blue area, but spends a lot of time in the blue area, then a lot of time in the black area, and so on. So when you look at these movies, the animals early on perform a lot of repetition. Sort of, they keep going over the same parts of the environment over and over. But eventually that is lost, and the animals get more efficient and start to traverse the entire labyrinth in a few seconds or a few minutes. And at that point, so this is after about an hour, at that point they routinely then are able to use the water port and use it quickly. So here's the same analysis of exploration after latent learning. So this is on day 2. The animal has experience of the labyrinth from day 1, and you can see that they immediately go into a mode where they cover the labyrinth quickly, and in the process discover the water port quickly and then are able to use it. Okay, so the upshot is that we think that there are 2 phases to learning of this complex task. The first phase is about learning the environment. It's like learning the structure of the task, if you want. In the case of the labyrinth, that means which location in space is connected to which other locations in space, and how can I get from the bottom left corner to the top right corner. So it's just about learning the structure of the problem. Yeah? If you think of other cognitive problems, like solving a math problem, there are certain techniques. There's a variety of techniques. At any moment, you could make different choices. Yeah? And with experience, you get a sense of which choices are connected to which other choices. Like if I use this method, then what can I do next, and so on. And so this experience of the space of possibilities is necessary to then quickly find a solution to any particular problem.
But part of the wheel, here you have red, blue, green, black in the left bottom.
Yeah.
And the fact that these are so clustered has to do also with the structure of your environment. So they explore one quadrant, then another quadrant, then a third, then a fourth. That's probably due to also to the structure of your specific environment.
Sure. Yeah, I mean, the structure, remember, is this hierarchical tree. Let's just go back, right? So
now
the structure is hierarchical.
Yeah.
So, it's easy to go back and forth between these 4 nodes. In order to get to these 4 nodes, you have to go out first and then come back in, etc.
Exactly. Right. So do you think they first create some local map and then they kind of stitch them together and get a global map?
Let me give some ideas for how we think it might work, and then we can talk later. But I think there is a hierarchy. I'm not going to have any direct evidence for it here, but we should talk about what sort of evidence you would like to see. Okay, so the sense is that there are really 2 phases to learning of this complex task. One I call structure learning, and the other I call solution learning. So the solution is the actual location of the water port and how to get back there. So how can you think about this mathematically? Since this is a kind of computational workshop, I thought I should have at least one slide about mathematics here. And so this is a suggestion for why this is necessary and how it might be implemented with neurons. Yeah. Okay, so very generally, many cognitive problems can be written as problems on a graph, search on a graph. Right, so the agent has, at any given point, in any given state, it has a variety of options. The options lead to different new states, and then within that new state, there's another option, and ultimately, you'd like to find your way to a solution. Right, so video games are just like that. Right, at any point in the game, you have a number of options. You choose one, then you have more options, etc. In the end, you want to win, beat the boss. Yeah. So let's suppose you have a graph like that, and let's suppose you know the solution is here. And someone drops you into the graph and says, go to the solution, go to the goal node. Yeah. What would be necessary to do that? Well, it would be great if you knew what the distance was to the solution, let's say this goal node here, from every other node in the graph. Yep. So for example, the distance from 1 to 2 is 2, because I can go here and then there. Yeah. The distance from 3 to 2 is just 1. I can take this step immediately. Yeah. So if I knew somehow, if I had a list, a table that says from every node what is the distance to the goal, then I can use that table to make decisions. Among the options I have, I just go to the option that has the shortest distance to the goal. From that state, I again take the option that has the shortest distance to the goal. Then I can kind of walk down the distance function to the goal node. This is a very common formulation, and it's a very popular problem in algorithms, how to find the shortest distance between any 2 nodes on the graph. Okay, so the animal walking around the labyrinth, it has information about local adjacencies, like it knows that I can go from node 2 to node 3, and therefore 2 and 3 have to be connected. There must be a corridor that leads from node 2 to node 3. And that's what we call the adjacencies. The adjacency matrix is basically a matrix that has a 1 in it when 2 nodes are connected by an edge on the graph, and a 0 when they're not connected. But we would like to get from this adjacency matrix, the local information, we would like to get global information. What is the shortest distance between any 2 pairs of points on the graph? As I said, this is an old problem in computer science and in algorithms, and there are many algorithms that solve this problem efficiently, but none of the conventional algorithms map onto the nervous system. There are things like the Dijkstra algorithm and the Floyd-Warshall algorithm, and all of them require complicated lists of actions that are taken and keeping track of 3 different nested loops. There's no way that you can build that out of neurons, as far as I can tell. So some time ago we asked, well, is there an alternative? Is there an alternative way to get all the pairwise distances on a graph? And we came across this interesting theorem. It says that if you know the adjacency matrix, then multiply it by some small number gamma, take 1 minus gamma, the adjacency matrix, invert it, and you'll get something that is essentially the distance matrix. it turns out that this, as gamma goes to 0, you can show that this goes to gamma to the minus d. D is the distance matrix between nodes I and j. And so what this says is that you can get all the pairwise distances on a graph by just doing one matrix inversion. There's no need for nested loops and so on. Now you might say, okay, matrix inversion is just as complicated. If you look up the algorithms for matrix inversion, they also have nested loops in them, so how would the nervous system ever do a matrix inversion? It turns out this particular inversion is trivial for a neural network, and this is how it works. So take the simplest recurrent neural network you can imagine. This is the one on, chapter 7 of the Diane and Abbott book. This is the one they start with. There's a single layer of neurons. The neurons get some input vector u. The output vector of the neurons I'll call v. And the neurons are recurrently connected. And the neurons just do something really simple. They take their input multiplied by gamma to make the output. Yeah. Okay, so you can write down the steady state solution from this network. The output is gamma times the input. The input is, of course, the external input that comes from u, plus the recurrent input that comes from the network matrix M, synaptic matrix M, times the output of all the units. And you do a little bit of shuffling of terms, and you find the answer is something times 1 minus gamma M to the minus 1 times the input. Right, so now this 1 minus gamma M to the minus 1 looks a lot like this here. 1 minus gamma alpha to the minus 1, gamma A, the adjacency matrix. So if we want such a network to learn this task of figuring out all the pairwise distances, we just have to ensure that the network matrix, the synaptic connectivity M, looks like the adjacency of the graph A. Well, here's how this can work. You just need to make sure that there's some Hebbian learning in this map network, in this M network, while the animal performs navigation in the, on the vector u. Let me say this again. Actually, let me just go through the steps here. Let's suppose a network like the one I just described. I'll call this population the map cells. They're recurrently connected to each other, but they also get input from another population of place cells. And the place cells fire when the animal is at a particular node on the graph. The agent is a particular node on the graph. So let's just look through this. Let's say the agent is here at X that excites this map cell because at this point the synapses have not been established. They are not, they're still at 0 strength. Only this map cell gets excited. But now the agent walks back and forth on this graph and excites other nearby neurons. And we're going to argue that by Hebbian learning, when the agent moves from here to there, these neurons are excited in close succession. I'll make these synapses, reinforce these synapses, strengthen them. And so after the agent walks around on the graph for some time, when it's now at position X, you get excitation of the corresponding map cell, but also map cells that are nearby in the network, where nearby means, with respect to the synaptic strength. And all of this comes about just from Hebbian learning within this network of synapses. You just make DDT of the synapse between neurons I and J somehow depend on the product of the outputs of the two neurons. And so if the agent walks from this place to that place, then transiently these two neurons are both high and they get the strengthened synapses. All right, so Hebbian learning in this network leads to a situation where the synaptic connectivity is proportional to the adjacency matrix of the graph, because two neurons that are place cells that are close to each other will often fire in close proximity as the animal traverses that link. And that's right, yeah. There's some exploration method that leads you through the whole graph. Yeah.
Also, you spend as much time on the different.
Yeah. I mean, you can make this saturate quickly so that it only takes a few links to actually produce a synapse. Anyway, so sometime later then, let's suppose that the animal finds this location Y, and this happens to be a reward location. Here, the water port is here. So that leads to, this extended activation within the map cell network, kind of a hump of activity near the place cell for Y. But now the animal experiences reward, so it gets to drink the water, gets dopamine from the VTA, and we postulate that dopamine leads to a stabilization or learning at this set of synapses here. And so the synaptic strength here, in some sense, stores the activity pattern that was in the map network at the time that the reward happened. So later on, when the animal goes back to some other location X, there'll be a different hump of activity here in the map network. And you can imagine taking the scalar product of this hump of activity with this stored synaptic strength pattern to get the excitation of the goal cell. And when this hump of activity down here matches the hump of activity during the previous reward, you get a strong excitation, whereas if this hump of activity is very different because the animal's at a distant location, you get only weak excitation, and you can show with a little bit of modeling that this translates into a goal signal that depends monotonically on the distance to the reward location. So after this Hebbian learning has taken place down here and this dopamine-triggered learning has taken place up here, this network now acts like a guide. It acts like a goal signal that can steer the animal to the correct location on the graph. The animal simply has to go among the options that it has. It goes to the option that maximizes this goal signal, and that leads it to towards the goal location in the graph. Yeah?
In realistic place, they also have something like appreciate fire rate in the neighboring location. So what's the main difference of this map cell with the
Good question, yeah. So in this model, it's a toy model, right? But in this model, the place cells are unique to a particular location, let's say a particular node of the graph in general. The map cells will eventually get response fields that are broader than those of the place cells, right? They have Well, it's interesting. If you look at hippocampal recordings, for example, CA1 or CA3, you find typically a range of sizes of place cells, right? There are some that are really tight, a few centimeters on a size in diameter, and others are broader, and there are some that reach all the way across the environment with some gradient. Yeah. And so we would suggest that maybe if the hippocampus is doing this, and this is a big question that I'll come to in a moment, that maybe the broader place cells are, the cells with broader response fields are these map cells, whereas the ones with tight response fields are the ones that are really just locked to the location in the environment. But again, the hippocampus, of course, is involved not only in solving spatial tasks, but much more generally tasks that can be written like a problem on a graph. And so, again, one would postulate, or the model would predict that there's a population of cells that has a broader response field than the original place cells. Yeah?
So I'm just wondering if the representation of the map cells are likely or instantly apart from one state to the goal location. Is that relative to a value function of each state, or is that not?
Yeah. You can take this model and mathematically show that it corresponds to some reinforcement learning models where this level here is essentially the model of the environment, right? The synapses here encode how the environment is connected. And this is the value function that is associated with each individual state after learning is complete.
Even though it might be very far physically. And that's a bit surprising, but it's testable, right? To really do it.
Yeah, I mean, the question then is, for any given environment, what is the proper abstraction into a graph?
Right, we have to make a decision. So when you have more than one possibility, you're not at an edge.
True. But, even in a long corridor, you still have the option of turning back. So there's always two possibilities. You could take the long corridor and parse it into many nodes, right? But this is a good question. Does the brain, what parsing does the brain ultimately develop for a problem like that? Let me move on to get some of the other questions that have come up already. So anyway, you take this rather simple structure of a neural network, and you find that it has, in principle, this ability to separately learn the structure of the environment through this map network, and in a different set of synapses, learn the location of any given reward in that environment. Yeah. I think of it, coming from Los Angeles, I think of this a bit like, a street map, and this marks all the Hollywood stars on the street map, like the homes of the stars. But of course, you can use the same street map to mark all kinds of other things. And the idea is that this network here serves a general function for this environment, whereas this particular network may well have a different goal cell for very different resources that appear at different places in the environment. So this is also a common network motif. If you look at the insect mushroom body, it's pretty much built this way. You have Kenyan cells with recurrent connectivity that's plastic. They output onto the mushroom body output neurons that are known to have their synapses modulated by input from a reward system. So it's very much that motif. You find a similar motif in the cerebellum, although people argue whether the right kind of plasticity happens there. Piriform cortex, hippocampus, so it's not a unique motif. It's something that's actually found quite prominently, yeah?
How do you know the environment? Even though you're going to be using Hebbian synapses, depending whether you're seeing the neurons are noisy or not, then it's going to be pretty slow. So how do you neural maps your experimental results in terms of the speed of learning?
The fact that one-shot learning can happen, I think, argues that this plasticity here is fast, that these synapses can be, in a single experience, can be updated.
The learning of environment.
Yeah. Well, in practice, this happens within an hour, but in that hour, the animal can traverse any one of these links hundreds of times, and so tens to hundreds of experiences of pairwise associations would lead to a synaptic strengthening.
So then it's not fast learning. So here you were saying at the beginning of the presentation that this is like a very fast learning, but in reality now you are saying that it involves like a lot of experience to get this
Yeah, I mean, again, to put real numbers on it, about 1 hour of experience. But that 1 hour might include hundreds of traversals of particular links. Okay, so the question is, can we tell where this is happening? If it is happening, where would it be? Yeah. And so, the mouse brain is a big place. It's only 1,000 times smaller than the human brain, but that's still a big place with, 100 million neurons or so. And so where should one look? And my students, of course, were eager to immediately put electrodes or miniscopes into the mouse head and record from here, record from there. And I argued, well, let's do some more experiments to see, can we say something about where this is happening, perhaps to steer better the electrophysiology. Okay, so here's a really crude experiment. We take a mutant mouse that is completely missing the dorsal forebrain. So here's a section of the mutant. This is a section of the normal mouse brain. You're well familiar with it. Here's a mutant mouse brain at about the same locations, a sagittal section. In the normal mouse brain, you have, of course, the basal ganglia. You have the hippocampal structures. You have all this beautiful cortex on top. And the mutant mouse is missing all that. This was created by Chris Walsh's lab at Harvard some years ago. As they were studying the development of the neocortex, they created this very interesting mutant. if you open up the skull, you're looking down straight onto the midbrain. There's nothing in your way. The olfactory bulbs are here. There's a little bit of flap of tissue attached to the olfactory bulbs. This is probably the evolutionarily more ancient piriform cortex, but all the dorsal structures are gone. Yeah. Now, in this first paper, the authors reported that, somewhat surprisingly, the animals survived, display relatively normal health, thus providing a unique animal model that distinguishes essential function of the cerebral cortex. So we decided to use that animal model and see what happens. A little bit of work was done to ask, is that acortical brain really, does it look like the normal brain except for absence of the cortex? And that seems to pan out quite well. So you can ask yourself, what happened to all that volume in the acortical brain that used to be occupied by cortex? Well, it's partially compensated by a flatter skull, so they really have a smaller head, and then there's a lot of water in the volume that used to be occupied by cortex. We took some measurements of subcortical structures that we know are important, like in the midbrain, and they seem to have the same size as in the normal animal. There's still a lot left to do to explore the anatomy of this mouse, but I want to just give you a sense that it's actually worth doing. Okay, so here are some movies of the acortical mouse exploring the labyrinth. This is after over an hour in the labyrinth, it discovers the reward port for the first time. And again, I told you there's a delay, so it can't come back for another drop of water right away. But here, sometimes later, this is now at 65, the 65th reward. The animal does these straight trajectories to the water port and back out. Yeah. If you look at the question of one-shot learning, they have the same ability to make use of a single experience. So here's an example where the animal discovered the water port after over 4,000 seconds. And the next time it entered the labyrinth from the cage, it took only, I don't even know what that is on that scale, maybe a minute to get there. Here's examples from another couple animals. But what you'll notice is that this number, 4,000, is much bigger than it was for normal animals, where it was on the order of 500 or 1,000. Yeah. So they take a lot longer to discover the water port, and when they do, they can use it. So here's an example of the timelines. I showed you this timeline before, where every dot is a water reward. And so here are 4 normal mice, and they explore the labyrinth and eventually find the water port and exploit it routinely after an hour or so. This is an hour. But you look at the mutants, they take much longer, 5 hours. 4 or 5 hours. Eventually, they can exploit it, exploit the water port location routinely. And so we looked at the exploration patterns of these animals. And so here's the time course for a normal animal. This is, again, plotting the nodes as a function of time. And remember, the different quadrants have different colors of the labyrinth. And so the animal starts by exploring locally, and then very quickly, after 2,000 seconds or so, it enters this more efficient mode where it covers the labyrinth very quickly, discovers the water port, which is this line here, and then uses it routinely.
Between the 2 successive results, we don't know if the mouse is just staying nearby or going far, right?
Yes, it just can't go to another end node.
Right, but we don't know if it's safe. And the thing which is surprising with the mutants is that after they repeat many water ports, then there's a delay where nothing happens. So it's as if they stay locally, and if they move far away, they kind of forget to it and doesn't have to rediscover it.
Possible.
But you know if those stretches where they keep, where the red dots are nearby are because they stay locally, or you also have cases of the mutant that go far but come back very quickly?
No, these tend to be just repetitive.
No, I'm talking about the top graph. You see, the thing is that the difference between the normal and mutant is also that the mutant, after they take the water 15 times, then suddenly there's a huge gap.
Yeah.
And so it suggests that they're exploring, they stay locally, and then when they leave, they forgot where it was and have to rediscover it. But
Yeah.
have you checked that?
Not directly, but that's a good question. Yeah. I mean, it can come out of analysis of these trajectories, of course. Yeah. But the point is that the acortical animal, despite not having a cortex or a hippocampus, it eventually settles into a mode where it covers the labyrinth efficiently and discovers the water port and can use it. So, of course, when we reported this, well, we haven't actually published it yet, but when I reported this at talks, people immediately say, oh, the mutant is such a different species. It grew up without the dorsal forebrain, so it probably does everything differently from the wild-type mouse, and you can't draw any conclusions from this, and so on. So the answer is, let's use a wild-type mouse, a normal mouse, and perturb its brain and see what the effects are. Yeah? And in particular, we perturb the hippocampus. Yeah? And so the way this was done is by injecting into the hippocampus AAV with this chemogenetic construct. So this is an effector that takes a ligand, a synthetic ligand, and silences the neurons in which the effector is expressed. And we separately, recorded activity of neurons to confirm that, in fact, most of the neurons in the hippocampus are silenced on these conditions. You can see that my student, with multiple injections, was able to really cover the entirety of the hippocampus with this construct. And so what's the effect of this? All right, let's look at examples again of these exploration trajectories. So here's a normal animal that settles into efficient exploration after 2,000 seconds or so. If you silence the hippocampus and do the same experiment, you get this very repetitive behavior again that lasts for a long time, maybe up to 10,000 seconds, before it settles into a more normal exploration pattern. Now, if you do the same thing on day 2, so the animal on day 1 had this experience. In our picture, it actually was able to do the structure learning correctly. And so now you silence the hippocampus on day 2, there's no difference from the normal animal. So the hippocampus on day 2 is not necessary to either recall the memory of the structure of the labyrinth, or to find the water port, or to remember where the water port is, or to recall where the water port is in order to exploit it again. So after structure learning is complete, the animals without hippocampus are working entirely normally. During? No, we haven't done that yet. But it's a good question, but there's no sleep in these experiments that I'm showing you here. So whatever consolidation is necessary can happen while the animals are actively exploring the environment. Okay, so just a summary of these hippocampal results that I find kind of puzzling. If you silence the hippocampus on day 1 when the animal is naive, it takes about 5 times longer to settle into an efficient exploration pattern where it can then perform this one-shot learning of the solution. And if you look at the acortical animal, the effect is very similar, maybe a little bigger, but the acortical animals have the same deficit, this long stretch of repetitive behavior. Well, what I'm going to suggest is that the role of the hippocampus here is actually in regulating the exploratory behavior. It's not needed to learn from the exploratory behavior or to learn the solution that comes after you've found the structure of the... It's totally reminiscent of your first slide or second slide, where the role of the anterior forebrain loop was in injecting signals into the system that allows it to explore and try out different options and find out which is better. I'll get back to that in a bit. Okay, let me leave you with something else that's interesting. You might think that if the animals acquire this memory so quickly, within an hour, then maybe it also disappears quickly. Maybe they forget about the labyrinth as soon as they go back home to the animal facility. It's not the case. So it turns out that this memory they have of the maze can last for many months. We now have tracked animals for 9 months without intervening experience, and I'll show you in a moment that they have a very clear memory of the maze. Here's just an example from one animal. This is the naive animal, and I haven't shown you this kind of plot before, but it's pretty simple. Horizontal axis is the time spent in the maze, and each of these green dots is the occurrence of one water reward, and I'm just showing them to you cumulatively, so you can kind of see that the animal didn't get any rewards, but then started collecting at almost the maximal possible rate after some time, like in this case, 1,500 seconds. There's another event here that we showed that the animals can also learn other things in the labyrinth. For example, how to very quickly get from one corner of the labyrinth to the opposite corner, the maximal possible distance. And again, that has sort of a discrete learning curve where nothing happens for a while and then suddenly the animal understands how to do that. Okay, now look at the animal after a 16-week period of no experience with the labyrinth, so just in the animal facility, boring mouse cage, nothing interesting going on. It starts to exploit immediately. As soon as you put it back into the labyrinth, it collects the water rewards, and it also does these very long routes through the labyrinth immediately. So there's some clear memory of this labyrinth that has been retained. Here's just more data. By now we have 30 or so mice that have been tracked for long periods of time. I'm showing you the length it takes for the water run, like how many decisions in order to get the first water reward. It goes down by a factor of 3 from naive even after 3 months. And they continue to learn. If you expose them every 3 months, they still add to the knowledge over a year or so. I should say that the mouse in the wild lives somewhere between 6 months and 18 months. So I really call this lifelong learning. It's a bit like the child learning to ride a bicycle. If the mouse learns it at age 4 weeks, it retains it throughout the natural lifespan of Mus musculus in the wild.
But then here it becomes even better.
Subsequent experiences can add, yeah. Okay, so one question is, what do they remember over 3 months? Do they just remember the structure of the environment, or do they remember the actual location of the water port? And so one way you can get at that is ask, well, what does the animal do the very first time you put it back in the labyrinth after 3 months? Does it go to the water port, or does it meander around? So recall that this is the trajectory to the water port. This is what an expert animal does. And we're going to ask, what are the decisions it makes at this location, and that location? And does it make the correct decisions, namely going to the water port or not? These are somewhat painful experiments because in this case, 54 mice. You get to use every mouse only once, not one day, just one entry into the labyrinth. That you want to know, what does it know about the labyrinth before it has any opportunity to explore it again. Of course, we use these mice for other purposes as well, but it's nice to be able to compile this here. Okay, so what do these decisions look like? First, here's a decision of a naive animal on day 1. First exposure to the labyrinth, never seen a labyrinth before, first entry. On average, over these 54 mice, they have some bias for which way they're going to turn. So, for example, on the first intersection here, they tend to turn right with some bias. And on the next ones, they tend to turn in the wrong direction, so down here and over here. So there's some bias that the animals have. This is significant. We don't know why it is. Animals come in with some bias over 54 mice. If you look on day 2, you find that these 2 decisions are strongly biased in the direction of the correct turn towards the water port. So this is day 2. It's just been one overnight period in the animal facility, and so over 54 mice, this bias is pretty strong and significant. So they have some knowledge of where the water port is. If you look at day 90, there's none of that remaining. All the decisions are 50-50. So the impression is that What the animals retain over these long periods of time is actually the structure memory, the structure of the environment, how things are connected in the labyrinth, and not the specific solution. And having that structure memory allows them to very rapidly traverse the environment and find a solution and connect it to exploitation.
So they make wrong decision and first time wrong, too bad?
Sometimes they do, but we don't count that. We just want to know what's their first instinct. When they first encounter that node, what do they do? Certainly, sometimes the animal turns right here and then comes back, but we just want to know what was the first decision. There are a lot of trajectory data, and you can analyze them in different ways. We just wanted to be very conservative here to ask, do the animals remember something concrete, the concrete solution, or just something about the structure? Okay, well, let me end here then and just summarize briefly. First of all, when you allow the animals to behave naturally, they start to look very intelligent. They look more like real animals than like cogs in a machine for alternative force choice experimenting. Under their own operant behavior, they can show you that they can learn 10 bits of information in 1 hour. We think that there are 2 phases of learning in these tasks. There's learning about the structure, which takes about an hour. Of course, a smaller labyrinth will take less, but anyway, in these experiments. And that's independent of any extrinsic reward. It happens even when the animals are just allowed to explore the environment without any rewards. And then there's the learning the solution, which is in this case the specific location of the reward node. And that's fast. That can happen after 1 experience if the structure learning has happened previously. The role of hippocampus, as far as we can tell, is to speed up the structure learning process. And it speeds it up by a factor of 5 or so by making the exploration more efficient. It prevents the animals from this excessive repetition of the same motif over and over, and instead allows them to explore more. Interestingly, we don't think the hippocampus is required at all for learning the solution, nor for recalling it after it's been learned. And then I showed you that the structure memory can actually last a long time. So wherever in the brain this happens, there is a long-term storage of this. So I want to finish with something I mentioned a moment ago, namely this idea that maybe the role of the hippocampus in learning, or at least in this kind of learning, is to guide the exploratory behavior. Maybe by injecting some randomness and allowing the animal to break out of a repetitive loop into something more productive that covers the environment better, allows it to see the whole graph environment and discover the solution. And of course, there's a huge literature that says that hippocampus is essential for the actual associative learning. This may not apply to all such cases, all such tasks, but if you go and look at some of the literature, some of the claims of evidence for hippocampal necessity, it may just be that the animals learn slower by a factor of 5, and the experimenters gave up and said the animal can't learn it. And this is a particular problem in the case of the Morris water maze, which is a key early attempt at experimental neuroscience to invoke hippocampal necessity, because you can't allow the animal to swim forever. It's going to drown. So if a rat in the Morris water maze keeps repetitively cycling around the same loop, eventually the experimenter has to take it out and put it back on dry land. So if an animal just happens to require 5 times longer to get used to this environment and explore it more efficiently, we wouldn't know on the basis of those studies. So you really have to ask, is there an absolute condition for the hippocampus being involved in learning, or is it just that the animals take longer and the experimenters become impatient? Anyway, I wanted to throw this out for discussion, because obviously it's going to lead to some complaints. Just a couple of editorial remarks at the end, kind of a little bit of how I see that animal behavioral neuroscience developed. 100 years ago or so, people were really willing to deal with the complexity of animal behavior, and so they built real mazes. In fact, this early rat maze was designed to be identical in structure to these Victorian gardens and some British castle. Sometime later, Tolman said, well, let's just make this whole the structure much simpler. Let's make the maze that consists only of T-junctions, and it doesn't have any loops. And so the only elements here are T-junctions and blinds. And then later Tolman said, well, fundamentally, the decision point is really the T-junction itself. Let's just work with one T-junction and make the animal go left or right, depending on some stimulus. And then when it comes out here, we'll carry it back over here and start it again. So we have kind of identified the element of decision-making. And then later people said, well, this is too tedious to pick up the animal here and bring it back over here. Let's just nail the animal down with a head clamp so it can't go anywhere. And instead of making it go someplace, we'll just put it into this paradigm of two alternative forced choice behavior where there are cues that start each trial and so on. Yeah. So obviously, this is very efficient. In terms of controlling the animal and having full control of every parameter of the task. But my sense is that something got lost in the process. And there's just too much reductionism. And that we started out with a behavior that's actually fascinating and where the animals are quite impressive in what they do, and it's puzzling how they can do it so fast and so on. But if you follow the standard scientific recipe for reductionism, you end up here, and this animal just doesn't do anything interesting anymore. It can barely perform a one-bit task after 10,000 trials. Let me remind you, it takes 6 weeks to train this animal to this poor performance, and in 6 weeks, a mouse in the wild goes all the way from being a baby to having its own babies. Yeah? So obviously it has to make a lot of decisions along the way, but none of them benefit from 10,000 trials of practicing anything. Yeah? So I just feel we've kind of lost touch with what animals are really doing, and there's an argument, I think, to return to this complexity of natural behaviors. Also, we should remember that we are the intelligent ones. So our job is to learn the mouse's language, not the other way around. We shouldn't force it to learn our language that comes from monkey experiments in the 1980s. Instead, our job is to learn their language and to use the fantastic tools we have from machine learning and artificial intelligence to try and figure out what they're doing when they're doing it, not ask them to perform to our standards. Yeah. Anyway, let me end here and just thank some of the people who are involved, Matt and Tony, the early work on labyrinth learning, and Ziyu and Zhang and Jiu have been doing some of the recent experiments I talked about, and Dan and Ruggieri are involved in other ways, and most of this was funded by the Simons Foundation. Thank you. Yeah.
Yeah, as you speculate, I will start with some complaints about the claim that Hebbian is not involved in learning anything here. I agree with you. It doesn't look like Hebbian is involved in learning the solution or the action sequences in this task. But why don't you think Hebbian is involved in learning the structure of the maze in this task?
Well, it's involved certainly in the sense that with a hippocampus, the animals do that 5 times faster. So this exploration becomes much more efficient, and as a result, the animals are able to exploit then the solution sooner than if they didn't have a hippocampus. On the other hand, even without the hippocampus, eventually the animals come to understanding the structure of the labyrinth, but it might take 5 hours instead of 1 hour. So I would say the hippocampus is not necessary for this process to take place, but it does accelerate the process by a lot. And of course, if you're a mouse in the wild, it can really help to have an efficient exploration mechanism that avoids doing repetitive movements all the time. So that's not I would say it's not a minor contribution. It's an important contribution, but I would say there's no logical necessity for the hippocampus if you allow the animal more time.
But is that possible that you might that doesn't have a different new cortex from the very beginning from birth, right? And some other system can compensate, right? Like I'm wondering if you have tried training a wild type mice that only silent hippocampus after they learn the structure. Do they still maintain the representational or memory of that structure?
Oh yeah,
or not?
yeah. I think we did that. Yeah. Let's see. This was. I know this all goes by kind of fast, so.
Sorry if I missed that part.
Okay, so this was the experiment I mentioned, where the animals are allowed to learn the labyrinth under normal conditions on day 1, and then on day 2 we bring them back and silence the hippocampus, and they immediately start with efficient exploration and also start exploiting the water port here.
Sorry, I understand too. When I talk about this, I saw those group of animals were silent both on day 1 and day 2, and you are constantly, so it's only on day 2.
Yeah, I need to make this more clear. Yeah, yeah, that's right. Yeah.
So like in previous integration papers, we kind of show that if you suddenly open a shortcut after not through the environment, that will take the shortcut to the next tile. And in your case, you can also actually just open a hole in the page which create a shortcut. And I'm curious whether the mouse will take the shortcut. And secondly, I think in the model, if there's a new shortcut which come out, you actually take longer to learn the habit. So I don't know.
Yeah. No, that's a good suggestion for experiment. Let me make the argument though that the mice are naturally learning and using shortcuts. So okay, so if we look at this. The animal, this was a trajectory by which the animal got to the water port the very first time from the home cage to the water. And you can see it's a very complicated trajectory. It probably covered this many, many times. And then came over here and did something other complicated down here first, and then eventually got to the water port. Yeah? But the next time, the animal cuts out all this stuff and cuts out all this here, and instead takes a right-hand turn, a left-hand turn, and a little jog here, but goes to the water port. So you can think of this as a shortcut, because the only trajectory the animal had known previously involved all this stuff here, and it cut that out. Yeah? So it's not that we opened a new door, but the animal saw that instead of going left and doing all this, that was my preceding history of trajectory, I can cut that out and shortcut.
Yeah, but I'm very curious, for example, in the shortest routine, if you open a door at the first made choice point,
Yeah.
then probably according to previous papers, they will critique, they will take this door and then go to the shortcut. But I think from the modal perspective, then in your neighbor matrix, you actually need to relearn that there's an impact.
That's true. Yeah. In that simple model, the animal would first have to traverse the shortcut to know that it's there, and then that can lead to a synaptic change that then reconfigures the map. That's correct, yeah. So I think experiments like that can distinguish this simple model, kind of feed-forward model, from one where there is planning of routes and so on. I should say that the classic evidence for this shortcutting from Honzig and Tolman's experiments is kind of weak. Like, if we had to review that paper today, we would have a lot of complaints. So, but anyway, it's obviously cited very frequently. And interestingly, it hasn't been replicated a lot, I should say. It's you would think that it's such a remarkable result that there would be a lot of literature following up on it. Yeah.
So I fully agree with this reduction approach, but I'm wondering what the fundamental boundary between this path learning and slow learning, because I think the machinery you propose like behavior learning or rapid learning, that could also work in simple tasks. But somehow that system is not triggered in these simple tasks, otherwise I can explain about simple idea of tasks. I wonder if you have any comments on like what really fundamentally different between this
Yeah.
heuristic task and the simple task.
Well, it's just speculation, but that's what we're here for. So, one aspect I think is that this spatial navigation behavior is very natural to the animal. It is used to, its genome has encoded probably machinery for making left and right turn decisions and then evaluating outcomes against those decisions and so on. Yeah. In the IBL task, the relationship between stimuli and responses is completely abstract and really dissociated from anything the animal experiences in real life. So the business that, if I turn the steering wheel this way, then the stimulus will move towards the center, and now it depends on whether it's oriented this way and that way. It's, I would say it's an association between stimuli and actions that is so unusual that it may, just for that reason alone, take a very long time to learn. A Bayesian would say that the animal's prior probability that this action is connected to getting juice is incredibly low because it doesn't match any priors in the natural world. And so if the prior likelihood of that hypothesis is 1 part in 1,000, then it takes a lot of evidence to overcome that very weak prior and convince the animal that, yes, this is actually what's going on. One should also remember that the animal, of course, gets lots of other sensory inputs all the time. We know that they're constantly moving their face around different ways. There are places on the skin that are itching probably, and so there are many possible associations between sensory events and the actions and reward that the animal could explore. So the prior likelihood that it puts on any of these associations, I think, is an important factor in how fast learning is going to happen. Yeah?
What if the ring gets larger and larger? So in this experiment, the maze is very more. It's being larger and larger.
I don't know yet. We've worked with smaller mazes, and so the period of exploration is faster, as you might expect. You cover less territory, but it would be interesting to see what happens with the larger spaces or so more complex spaces. So we've also played with a very different kind of maze that allows you to very rapidly reconfigure the structure of the space to understand better how the structured learning works. And in particular, there are mazes you can build where there are a lot of loops, so a lot of alternative pathways to get to the same goal. There are also lots of ways of getting stuck. And I think those are going to be quite informative about these processes of structural learning. But I don't have anything clear to say yet. Yeah? Well, I mean, one option is, again, getting back to Adrian's slides. I should have asked you for a copy of the slide because it just explains all this stuff. Anyway, so, Adrian argued that the synaptic learning is at the level of effectively the basal ganglia, but it is affected by signals that come from the anterior forebrain loop that inject variability into the process. And so perhaps the circuit that's actually doing the learning, the stimulus action association, is closer to the basal ganglia, but it benefits from inputs from the hippocampus and cortex in the process of exploration, the process of learning. Even though the learning itself maybe is happening there, the actual synaptic changes that make the association. It's basal ganglia obviously is part of the brain that still exists in the acortical animal. We're talking about trying to trim that animal, that brain back further to see at what point you really disrupt this ability to learn. Yeah.
One is comment earlier on the typical training of the idea of task. I think the main reason for how to train is because of that design of that task. The similar to that design, the normal quality that is designed. So if you make the report very straightforward by looking to them more, and if the stimulus is like easy like older or sun in your case, and if you make the environment very clean, then it takes very few days, and sometimes within 3 days for older it's faster.
Yeah.
It's just their task is for the design.
Yeah.
That's the problem.
I think it gets at this issue that both the stimulus and action variables are not part of the natural repertoire. But if you make them part of the natural repertoire, like food odors and actions associated with consuming food, like licking or digging or so, then you can get much faster learning rates.
So if understood. So sorry, if you don't mind asking.
Let's welcome Haiping again for his advanced lecture, Non-Hopfieldian Dynamic Challenges Opportunities. Thanks, Haiping.
Thank you for the introduction, and now come to the advanced level. And I should have a recap of the previous lecture. We talk about the computation, from computation to theory and mechanism, and to algorithm and hypothesis. And this style, I think I love this style very much because if you put in the Hopfield style computation, it means that you construct the coupling as a Hebbian rule, and then you can make the network to carry out some memory recalls. But if you use the statistical mechanics, and then you can reduce this high dimension system to one-dimensional recursive curve y equal to fy. If you look at the Hopfield, the model, we have retrieved all the phase diagram or the memory retrieve phase from this single diagram. You can look at the Hamsprinsky paper, and then based on this, and then you can extend to the modern machine learning. We call the mode-based training. You just generalize this as a Haiping coupling to the multiplication of the three matrices. You apply on the supervised learning and the spiking neural network, and even you can include a low-range representation of the neural computation. So this is a paradigm we talked about in the previous lecture, the basic lecture. Now we want to go into the beyond the I call the non-Hopfieldian dynamics. And now the JIJ is, for example, if from the computation side, we know has reservoir computing developed 20 years ago, and people say that the JJ can be independently sampling from the Gaussian distribution, and then we have a G is the variance is the synapse gain. If you change the synapse gain, and in this network there will occur a self-organization to high-dimensional chaos, and this is developed in the 1988, maybe about 40 years ago, and now if we go in from this paradigm that is parallel to the Hopfield network, in the non-Hopfield network, what we know in the theory of mechanism, or can we develop some algorithm and generate some testable hypothesis for the experimenter? And then this is what we talk in this is advanced lecture. So I think we start from I think one student of the John Carmel here. In 5 years ago, 6 years ago, I invite Carmel to our university to give a talk, and I think the chaos is very beautiful. And I think in the nature, because chaos theory also captures the minds of many men and women in art and science due to its ability to demonstrate how complexity can arise from the simplicity. I think the complexity out of the simplicity is our general belief, even when we're doing brain science or doing machine learning, and this is our basic law. This is what we talk about the theory of mechanism, because we can understand complexity based on some simple rules. So I think now we come back to the classical paper in 1988. I think this paper generates now, I think, the basic framework of the theoretical neuroscience, because you want the understanding, we use some theoretical tool. And this paper is called Chaos in Random Neural Network. I think it has four big contributions to our community. The first is mathematical to illustrate why the brain addicts stochastic behavior. And the second one is a dynamical transition to chaos. This is a high-dimensional chaos, and in the mathematics and physics, there's never had such kind of model before 1988, and also the connection from the disorder to chaos, because the coupling. The diagram may be very random. Of course, it is structurally random. But whether this kind of disorder can lead to the emotional behavior of the neural network, then this model gives you the first example. And also in theory, they link the dynamical mean field theory to chaos. But in nowadays, if you look at the theoretical neuroscience paper, if you use theory, I think this dynamical theory is the first choice. And then all this is for contribution is established in these papers. And they can reduce this high-dimensional system to one-dimensional system dynamics. This we call the Newton dynamics. Please remember, Newton dynamics, their form can exist in our recurrent neural network. So then, of course, then you should change the coordinates of your state. The coordinate now, it is a correlation function. So in our original recurrent neural network, the state activity, of course, not the Newton dynamics, but their correlation becomes Newton, and there's a potential. And then use this potential, you can determine the transition every 4 elements of this is the mean field theory. But I will not talk the detail of here. It will be many classical introduction here. But in this lecture, I just challenged this one-dimensional theory, and also we can look at the one feature of the chaos, and then if you want to predict the activity of each neuron, it could not be done in very long term. It's just very short term. After that, even you can learn very precise structure of the neural network. What I mean precise, it means that the numerical accuracy may be within 10 to minus 8 or 10 to minus 12. It is just a little bit deviation, but the deviation can be amplified if you want to predict the trajectory of the neural activity. So it just has a cutoff here. So this is due to the chaos, right? So in this talk, I would provide a new theory about the high-dimensional chaos. So I just look back to some dynamical system theory. I think in nowadays, if you use a dynamical system to equation to describe the neural activity, you can write the ODE, right? The ordinal difference equation, and this we call the speed, right? And depending on some extra parameter and some at the activity itself, right? For example, the extra parameter can be coupling. Right, can be the connections or some external stimulus, and then you can study the volume of phase space of this dynamic system, and then you use some mathematical theory, and then you can study how the volume changes the relative speed, and then you can write up as a divergence of the external force, the divergence. This is called if the divergence is become negative, we call the dynamical system is dissipative, right? Of course, our neurodynamics is always dissipative because they are focused on narrow down into the very tiny region of the phase space. So this is if you run in the dynamics, it is dissipative dynamics. This will give a little bit mathematical background. But originally, from mathematics, the people very interested in the dynamic system from Poincaré, maybe more than 100 years ago, because they think about they want to be structured to characterize the whole space, how the whole space look like. This is a large scale or global structure. It determines the function of each neural trajectory. But even until today, until the 2023, people still struggle to understanding this kind of the chaotic neural network. For example, the unstable fixed point. If you go to chaos, I think all the fixed point it becomes unstable, and how they rely and how the relation with the neural function. So this group of people try to focus on a global geometry of the fixed point, and the basic equation is we already very familiar in this is in this summer school, many people talking about this kind of the structures. I did not talk about the mathematics, but we just focus on their results. So it gives us a very surprising result. For example, they can calculate the complexity, that is the number of the fixed point. And then after the chaos, right? Chaos it happened here, and then the number of the unstable fixed point it is exponentially growing up, and very surprising means that if you compare the dynamics and the fixed point, they have the separation on the different places in the phase space. This is a form of sphere, a very thin sphere. The dynamics is in a lower part, but no, the fixed point is on lower part, but the dynamics is the upper part. So it means that now we have a gap between the dynamics and the fixed point. So is there some mathematics too or physics picture about this is a global structure? So this is the topic of my talk here. So if we look at some low dimensional system, you can visualize it, but high dimensional system you cannot visualize because it's so what we call it is challenging for us, right, to understanding the neural dynamics. But low dimensional system, for example, this is I think this is 2-dimensional or 3-dimensional. It's 3-dimensional view. You can find the green part is the fixed point, right? Because this is dimension is so low, but the fixed point I think is countable, right? Because it's 3, 4, 5, 6, and something. And then you can find the blue one is the dynamics. So the dynamics is the fixed point is surrounded by the dynamics. Right? It is get close, but it appear a very specific structures here. So I think the main idea this means that there's some hidden relationship between the dynamics and the fixed point. Even the fixed point is unstable, but it is useful. And the people in the field, it is rarely explore this part nowadays. So we want to start to put some theory or mechanism about this and try to think about whether can we generate some testable hypothesis for this non-Hopfieldian dynamics. Because I should emphasize, if you use the Hopfield, and then this dynamics will flow to the fixed point. Then for each fixed point, we call the memory, right, or even the attractor. But in this kind of the network, because the JIJ is the coupling is not symmetry, and then the fixed point cannot be approached. So this is a huge difference from the Hopfield dynamics. But here I call the non-Hopfield dynamics.
Whether chaos exists in whatever non-Hopfieldian in your
but it will become very complex. But I think if you have a non-symmetry one, it has a possibility to generate chaos. I say it has a probability, but of course you can for some certain network you just have a fixed point. It is not chaos. So it depends on your
definition
setting. It still depends on your setting. I cannot say definitely.
of non-Hopfield is just
maybe a necessary condition.
Usually way to do that is try to plot the Lorentz map according to some parameter, like what the fixed point will change according to the parameter change, right?
Yeah.
And categorize that. But here, it seems you don't consider the change of some parameters in the dynamics.
Later I will actually we just change one parameter to generate the high dimensional chaos. I will detail that. I think this is based on this paper in the 2014. It's almost 1 year later. 1 year earlier, I derived a new kind of the angle or perspective to look at the high dimensional chaos. But we are very lucky and can connect so many concepts. For example, self-organization, criticality, and edge of chaos, and response, and unstable fixed point, and more. I will talk about how to link this kind of different concepts into within the single frameworks. Now we make a toy model because physics or mathematics are always working on toy model. We have the brain, right? The many neurons, and then we just use a set of ODE, and we run, because for each neuron they receive the synapse currents from this neighboring neuron, and then this is their JIJ. This is a synapse, it's a symmetry, and then for each neural activity you want to contribute to their synapse current, it should be go transfer from by nonlinear function. This we call the activation function, and then this is a nonlinearity, and then sum up, and then you give a currents. But this is a decay term, right? Because you make the synapse currents, they change over time. So this is a decay term. If without the JIJ, of course, it decay to 0. Of course, you can put a time scale here, and time scale can depend on each neuron. But for simplicity, I just assume a time scale equal to 1. But the time scale doesn't affect our following analysis. But we just use this equation. Why all the theoretical neuroscience paper favor this kind of the setting? Because they cover the 3 characteristics. First is asymmetric. Even I can put the correlation between synapse, and second point is nonlinear dynamics, and 3 point is non-gradient dynamics. Non-gradient dynamics, it is in contrast to the Hopfield. Hopfield is gradient dynamics, but here is non-gradient. If you cannot find any perturbations, right? If on this is ODE. So now we want to get close to biological detail, but it is not very exactly the biological detail, but we get close to that. And then you use G to connect the variance of the JIJ because it is random coupling. That is what we use in the regular computing. And also we can put some correlation because this neuron connecting to its neighbor, and the neighbor send back his synapse to this to the neuron itself. I think this both direction can be asymmetry, right? It can be independently adjusted. So we put the correlation level characterized by the gamma, right? And if gamma equal to 1, of course, it is a symmetric network because Jij equal to JJI, because Jij is from J to I, JJI is from I to J. So it is the same one. But if it can characterize the anti-correlation. Anti-correlation, it means that Jij equal to minus JJI, and then gamma turns to -1. So I use I can tune the gamma from -1 to 1, and then cover the whole spectrum of the correlation level for each direction of the single connections. So from there, the G and the gamma is our main the key parameter. Sorry.
So what is the transfer function that you use here? Is that sigmoid?
It can be arbitrary, but here I just use the tangent edge. It depends. The activity depends on your the activation choice. Yeah.
So the question is,
It can.
Then, can you have a chaotic regime because it will explode?
Yeah, but here I just do Benson. It should be saturized.
Sorry.
It should be has a saturized activation function. Yeah. Yeah.
How realistic is this to model? Because in the traces that you have at the bottom, then you have saturation.
Yeah.
So this is the way you control. You can have chaotic regime.
Yeah. But it's bounded. It should be bounded and saturized.
So it's possible to have like a chaotic regime without reaching saturation.
Yeah, something like this. Yeah, we have never tried on other activation function, but here I recognize the one necessary property should be bounded. And because this tangent actually function is bounded, similarly also bounded.
How runs this in the brain? Because something.
Yeah, I never tried. Yeah, you never tried, but I never tried DEX. yeah. People, if interested, can try. But our theory can apply. I think it doesn't depend on the choice of the. But the phase diagram dependent, really dependent. But our theory doesn't depend on the activation function. Yes. Now, if you do the simulation, you can clearly define the two phases. One is a phase point. This is a trivial phase because the G is weak and it depends strongly on the G. If G is weak, and then even the gamma it goes to -1 to +1, and this area always fixes the phase point. So G is a synaptic gain. It's a key parameter. But if the G detuned a little bit larger and it's quite dependent on the gamma, the correlation, the law of the correlation come in, and then it will be generalized the chaotic fluctuation. So because this kind of system from this phase diagram is quite simple. It is just two phases, and then it has a critical line, and here this critical line can be analytically determined because for the phase point it becomes unstable with respect to some variation, and then you can use the Jacobi equation, Jacobi condition to determine this is an analytical line. This is a marginal line. That is a stable, a marginal stable the line of the fixed point. If this is line, if the cross this line and the fixed point becomes completely unstable, and then it will lead to the diversity in the neural activity. So this is a basic structure. For example, many people completely determine this. For example, from the Brunet's paper with the physical real E, 2018. But this is, I think, what we already know about this kind of picture. But here, I just to talk about from the dynamic system theory. If you think about the equilibrium limit, right? That this kind of the dynamics can be flow down to equilibrium, and then we already has one classical example that they put Jij equal to Jji, and then you for example you put a linear function, the choose activation function as a linear function, and then you can write this dynamics as a gradient dynamics. The Lyapunov function is very formula, I think, is statistical physics. This is something called Hilbert's Ising model and something like that. Of course, you know, Hopfield model it is born from the Ising model, and then you can draw this is energy potential, and then you can understand very well what the memory recall like, because you're starting from some crafted image, and then you will be, because this is a gradient dynamics, they will be come to the lower point, and then this lower point will recover what your mental image about the memory or something like that. And also from this kind of the system, if you look at this, of course, you can add some noise, then you turn some the Brown motion, and then you can know there has a stationary distribution if you formulate with the Fokker-Bracker equation, because this is statistical dynamics, and then it is likewise the Boltzmann distribution actually in the statistical physics. So we call this as an equilibrium problem. It is not out of equilibrium problem, and so this is we can guarantee a Lyapunov function decreasing over time, and this picture is completely mathematically clearly. But I think it is not very biological. We need non-gradient dynamics. If now we write the very complex arbitrary force function, and this force function cannot be write into a gradient term, and then you add some color noise or white noise. It depends on whether you want. And so this is much more general. I think you can apply on to map to fix or experimental data and a lot of something. But here, if you look at the theory, we know we can have the steady state, right? We can have a steady brain state when you're doing computation, right? And then you just put some assumption. You assume the steady state has some kind of form of the exponential family, right? And you have a non-equilibrium potential, something like this. This is only applied to the steady state, right? And then And then you use a Fokker-Planck equation. This is a probability conservation. This is a probability flow in the neurodynamics. It has a probability current, right? The divergence. And then you put this probability, because the steady state, the probability doesn't change over time, right? And then you put this 0, and then you can solve that this is a probability current for neurodynamics. And then you put the steady state and just to resolve this is f, re-express this f, all right? For example, this is j divided by p, and this term move to this side, and then you can interpret the force as a 3 term, right? The fourth term, I think it is a probability flow. So probability flow can have as a force. This is a curve force. Why is it curve force? Because the divergence free. So this is a curve force. It is something like that. It is like our magnetic field, right? The curve force. And this one, it is what you assume in the potential of the steady state. So it is like a gradient. So this actually has a gradient term here, right? But it is to be coupled with the curve force. If you do not have this curve force, and then the gradient will disappear. So it means that this f is arbitrary, the steady state will becomes very complex. And this, of course, this D is a diffusion matrix. It characterizes the strength of the statistical noise. So anyway, if you try to use this framework to understanding the arbitrary dynamical system, you should know the potential, you should construct the potential, but people do not know how to construct the potential. This is the problem, right? Of course, you can do some perturbation or something like that, but then if you go into the very detailed analysis of your data, and then you will cause many problems. And that is the probability even for statistically, it is generally unknown. So it means that this, if you want to study the non-equilibrium brain state, it's very challenging and very difficult. So now we propose another angle by ourselves, and we do not use this form. I do not. Sorry? Yeah. Non-equilibrium this. Yeah, later I will specify that. It's very challenging, and I don't talk about this and this. And now I propose a new idea. I proposed 1 year ago, and now we just look at we just construct an energy function, but this energy function we call the quasi-potential. And why we think about this, we just do some simple example. For example, 3 neurons, and there has a bidirectional connection, and this is asymmetry, and the nonlinear function is the tangent edge function. And then you observe that if the coupling is weak, and then there has a trivial fixed point here, it is just a fixed point, but you increase the strength of the connection, and then this is trivial point will be distable into the two focus, this dynamical system, because this is 3-dimensional system, it is no chaos. But if you look at carefully and you trace for each trajectory, and you will find the color changes. Why the color? Just focus on the speed, right? The key parameter is the speed, and this speed is very fast, and then the speed slower. Go to fixed point, the speed equal to 0. Right? This is speed true. If you look at the dynamics can be captured by ODE. So now we can write a scalar function. That is because you know the speed in the physics, we have the kinetic energy, right? So it just uses what you learn in the college physics is with kinetic energy equal to mv, right? This is the speed. Right? This is what you learn in the college of physics, for example, in the gas or something like that. And this one, and then we apply this concept to our neural network. And because neural network, you know the speed, right? If you write ODE, you know the speed. So we put the speed here, and this L2 norm, and then 1/2. But in the physics, we have the mass. But of course, our neural network, we do not have a mass, but we just set up it as a unit math. And this is a metaphor, right? Because what we care about is this is speed, the speed. So if we write out this, then you can use this scalar function to interpret every trajectory in the dynamical space, right? So here we call the non-equilibrium steady state. It means that the kinetic energy reach a stationary area, right? It can be it cannot be very large, but it should be has a certain level of fluctuates with it. But if the energy if equal to 0, we call the non-equilibrium fixed point. So we can characterize the two very important regions for the neural dynamics. But if the energy it changes rapidly, and this we call the transition dynamics. So you can cut the general dynamics into 3 stages. One, it is a transition, and then non-equilibrium steady, and then non-equilibrium fixed point. But the non-equilibrium fixed point is very strange. If for the chaos case, you cannot be reached because it's unstable, right? So you cannot be stay there. In this case, we just to select some phase space. So our idea is the phase space for any dynamics. It can be partitioned into different regions of different speeds. Then you look at this new picture. I think you do not need to assume some potential. You do not use the dynamical mean field theory, and then you can create a new way to understanding the general picture. So now, if you want, then we look at the neural dynamics as optimization of the energy, the kinetic energy, because when you run in the dynamics, you just to be make your speed as low as possible, because you could not be fast retraining, because it's not useful for computation. So now we now know the scalar function. We can just doing as a machine learning as a gradient descent and add up some y noise, and then we can create a new type of the neural dynamics, right? So then you know the scalar function. You can easily to do these gradients. We focus on its components. Then we write it explicitly. Then you have a surprising finding. If you write explicitly, they have a 3 term. Each force is a 3 term. To know that this HI, it is a synapse column. It's a weighted sum of synapse column. Maybe I will return back to here. The HI is here. In the last lecture, I write this term is HI because this is for neuron I. How large of the synapse column they receive from its neighboring neuron. I write this HI. So this is kinetic energy. This is minus SI plus HI, right? If you do the gradients minus SI plus HI, it's here because this is due to the kinetic energy, right? Because kinetic energy has a power 2 here. It is a speed up to power 2. So if you do gradients, of course, it is the nature should be has the first term. But the third term is very, very strange because in our previous recurrent neural network, we just only these 2 terms. We do not have the third term. But if you use this kind of the scalar kinetic energy perspective, you have the third term. So what the third term mean? So this is very clear because if you look at detail, right, and this is the slope of the activation function of the neuron I. So this term played by the nonlinearity of your dynamics, and this term is sum over J, and J it is a neighbor of the I. It's a neuron of the neighbor of I. And then you look at this term. This is coupling the JJI. It's not JIJ. It is a retransit, right? Maybe I will draw the diagram maybe for easy to understand it. For example, this one, originally you just collect your information from your neighbor, right? Neighbor neuron, right? This A, this is JJ. No, this is JIJ. This is from its neighboring. This is JIJ, right? JIJ it is from here. Then you sum up all this, and then we calculate his input current, right? But now you look at this third term. It is on the other way, right? Because now it is JIJ, right? JIJ it means that this I, because the connection it is a double direction, and then it must be going to here, right? This is JIJ. JJI. This is I from I to J. So from this neuron to impact its neighbor, right? So this is the impact on neighbors, not neighbors' impact on himself, right? So because this is both in our brain, maybe something, the connection is both bi-directional, right? So this is Jij. Then you look at this term, right? Hj minus Hj minus Xj. What is this? This is speed, right? We just look like our ODE. It is something like Si dot minus Xi minus plus Hi, right? So this term, it is we call the speed. The speed, right? So this term, it is a speed. So the speed is the neighboring speed weighted up by the other direction of the coupling, and then you will affect the neuron ice activity itself. So this is a new kind of the dynamics of the structures we found on this is a kinetical energy perspective. It never appeared in the previous study about the neurodynamics, right? Oh, eta. Oh, no, I don't know. Eta, it means that because the Professor Lobo says that if you use ReLU, it means that if you use ReLU, I think this term can be arbitrary because the ReLU can be shaped can be scaled by the neural activity. So this eta is just constrain the neural activity to the certain level to avoid arbitrary. But here, I use the tangent edge function. We do not need to this term because tangent function is bounded. It is a unit. So it depends on activation function. I think this is eta. It is a regularization of your activity. Okay, now sorry.
It's a very weird term, right?
Yeah, very weird term, but very powerful term. Later, I will explore.
Intuition for me, like it's kind of the force toward this element. It determines the force it exert to others. Never do that, right? We always say that neighbors impact only you.
Yeah. The point is it means that originally for each neuron you impact by its neighbor, right? Yeah. But the point is now for the neural activity it changes and then it will send out its feedback to its neighbor, and the neighbor will be affected back to the Xi. This is a feedback. So this is a feedback. Because this is an important character of dynamics. We call the feedback because it has some delay, and then it will come back to affect the neuron itself.
Overcounting the message you send to others, you want to delete that.
Yeah, delete. Yeah. Later I will show this term will stabilize the chaos.
If this term is a feedback, why the feedback not contained in the Jij?
Yeah, the feedback going to this way, right? So it means that you mean this is the feedback is I to J, right? The I to J, but its neighboring use the coupling from I to J. It is not use the J to I. This is a strange part, right? And also now this feedback term affects the neuron ice activity. It uses its speed. It do not use its activity. That is how rapidly changing of your neighboring, it will be feedback to the neuron and then to regulate the neural activity. So this is a very strange type. I do not know whether people can identify this effect in the experiment. The speed is important, but now it is the speed matter. It is not the activity that matter, because in traditional Hebbian coupling, everyone call the activities is matter, but now it's speed matter, not the activity. So now we can write down the force. Sorry.
I'm kind of confused. So the energy function is constructed from the speed in the original dynamical equation, but how come the speed calculated from the energy function at here is different from the original velocity?
Yes, speed because you can write down the ODE, and then the speed is the force and then square. Your force and the square. This is quite easy. If for ODE, every kind of ODE you can write this.
Yeah, but in the original ODE, the speed term only contains the h minus x, right?
Yeah, the original one I think it is here, right? Original one. Originally this is actually it is speed, right?
Yeah. Why did in the last page the f contains an extra term that is like a feedback term?
I think this is due to your definition of the kinetic energy, because there's a power 2 here, and then you do the derivative, and then you, of course you have the extra term, right? Because this derivative with respect to xi, right? Respect to xi, the first term, if you do the derivative first term, give the original term. Because this also has a xi's contribution, right? Because xi can affect the xj, and the xj can affect the xi again. This is a delay loop. So if you look at detail, I think this is very simple. You can find the xi here. For example, because this sum over I, right? If you look at this xj and then the xi, the law of xi will enter the synapse current. So when you're doing the derivative, you should consider this is a xi. In fact, is affect its neighbor, right? Because from this kind of dynamics, the xi can be a neighbor of xj, and xj can be a neighbor of xi. It is a mutual interactions for each kind of the neuron pool in the activity. So then it is naturally we have the extra term right here.
The way you define the n function, is that like the echo function or not in this case?
Sorry?
So the way you defined the previous slide, right? So you define kinetic energy. So would it be like some echo function or?
No, we cannot call this Lyapunov function. For example, if you write out this dynamics, dynamics of phi, it is completely different from our original dynamics. Because they have an extra term. If the extra term can be shut down, and then completely the original one, so it will be different. But there has one condition, and two dynamics is the same. It means that the speed, if it's speed equal to 0, and then this term shut down, and then it's just a fixed point. So these two time dynamics is share the same set of the non-equilibrium fixed point. Beyond that, it's completely different. Right, you should know this point. It only equivalent in the 0 speed, right? 0 speed because of course this term disappear. So now but the very nice case, now we have a gradient dynamics here, and then we know there has an exact stationary distribution from the focal point equation. It is a Boltzmann distribution, and then if a Boltzmann distribution, then we can use many tools from statistical physics to study for non-gradient dynamics, for example, order parameter or something, and you just need to tune the beta, right? If, for example, the beta, it is a statistical noise in the ODE, right? And then if you tune the beta tends to infinite, that is 0 temperature, right? If you tune the 0 temperature, and then if beta tends to infinity, and then the energy function should be 0, right? So be 0, it means that it will capture the non-equilibrium fixed point, and then this non-equilibrium point is shared by your original dynamics. So you can study this analytically tractable case, and then to study the intractable case of the originally problem, non-gradient dynamics problem. So then we can divide some order parameter, we can characterize the phase transition, and many amazing property will come out. Now sorry?
So here the probability is like the new dynamics, not the original one.
No, not original. Only in the statistical noise. If you take the GLO statistical noise, and then you will get approach the original problem, right? Because 0, if you take the 0 and the beta equal to infinite, if you according to Boltzmann distribution, the E should be 0. E should be 0, that is the fixed point of the original dynamics, right? If you look at our energy, if E equal to 0, you do not, because here we do not use this regularization. If E equal to 0, because this is a power 2, it should be all term of here should be 0. That is means in a traditional dynamical system analysis, the way you find the fixed point, that is let the derivative with respect time equal to 0, right? This is a fixed point set of the high dimensional system. We can use this measure to identify the fixed point for high dimensional system. But for the two dimensional system, of course, you can solve two equations, and then you can identify fixed point. But for the high dimensional, you have no way to identify the fixed point. But here, it is one possible way, acceptable way to identify all the fixed points. So we can look at the pictures about this. Now I just to think about a very famous saying about general relativity, because the fellow from the Princeton University, they say the space-time tells matter how to move, and matter tells space-time how to curve. So we can map in the similar general relativity to our brain. So brain is a brain connectivity, that is a coupling, and the coupling tells the brain state how to move, but the brain state will be through the synapse plasticity to affect the brain connectivity. So they interact with each other and give the emergency of the behavior or something, decision-making, perception, and can be generated from this kind of, so this is a similar general relativity. So you know, in our 3D space, we have the different mass, for example, sun and earth and a lot of things, but in our brain, we also have a different kind of brain state. It's not the unique state. So if you think about this brain state as some non-equilibrium fixed point or something like that, and then you can define the free energy, right? Because from the Boltzmann distribution, we know there's a partition function. If partition function, it is a general function, a generation function. You take the logarithm, and then it can be defined of free energy. But the problem now is the brain connectivity is random, right? If given one realization of brain connectivity, it can give you one free energy. But if you change your brain connectivity, they have the other free energy. So the free energy now becomes a random number. This is in contrast to traditional physics, because in the Ising model, the free energy is determined, right? But now the free energy depends on brain connection. So we should average out the brain connection. How to average out this brain connection? It becomes a mathematical appealing things, right? That, for example, now it is logarithm g. It is a free energy. Then you want to do this average, but this average our brain connection, right? Brain connection. But this is very challenging, because this is a logarithm nonlinear function, and the brain connection determine the z, but you tell logarithm. But actually, we have some mathematical identity. We just take a limit, right? Into 0, and then we gn, and then divide by n, and then we take a logarithm here. Right, logarithm gn divided by n. This is a hospital limit. You just doing the simple calculation from the college math, and you can prove this is exact, right? But the problem now, you have the partition function to up to n power. You treat this mathematics, right? But this one, I think, is very nature, because here we already know we have different kinds of brain states. So we can map one brain state to one n here, because n it is a power. You can be n is integral power, for example, 4 power, and then it means that you have a 4 brain state. And if it has a 10 power, and then have a 10 brain state. So each index can be assigned as a brain state. So you can look at our mathematical formula, and then there has because the partition function is integral out all different kinds of brain state, right? So now you can see our kinetic energy term here. But now for each state in our kinetic energy, we have a superscript has an index A. Index A is response from the power of the partition function. But the index A, it just has a direct meaning with the brain state, right? For example, you have one brain state, then you call the A equal to 1. But if the brain state changes to the other brain state, you can call equal to the A equal to 2, right? It never mind if your brain state is exponential, it's okay, because the N can be an integral number, right? Arbitrary integral number. So now, want to calculate the kinetic energy. Now the X has a superscript. It clarifies which brain state you want to address, and the lower and the subscript is called which neuron you address, because the neuron, the number of neurons is fixed, but it can appear on different kinds of brain state. So it has 2 index, right? So this is of course it is due to the mathematical the limit, right? But I can interpret as our brain state mapping. So actually this is correspond to some. Sorry.
Where's the N? Yeah, where's the N in the formula of CN?
Yeah, so in the lower.
There's no N here, right?
Here no A. Oh, here you mean this one?
Yeah.
Sum of A equal to 1 to n. This n is integral. This is integral number. So here we use this has a subscript and a superscript. Subscript is a neural index, and the subscript is a brain state index. You just count all possible brain state, integral out all possible state in your brain, right? So you can lead to a partition function. So I think this is what I call it is a replicate dynamic state, because this is power to n. That is, you had the n identical system, because each system contribute to one partition function. So this is a replicate dynamic state. But in physics we call the multiple equilibrium. That is I think it provided Giorgia Parisi, I think in the Nobel lecture in the multiple equilibrium in the 2024. So we can have the Parisi's pattern in the brain state, as you call the multiple equilibrium. But we know, of course, the equilibrium can be fixed point, can be non-equilibrium fixed point, even can be a chaotic or attractor, it can be arbitrary. So anyway, you can integrate different things into the system out. So then we can derive all kinds of the conclusion from this calculation. Of course, but this limit is weird, right? The integral number finally is be 10 to 0. So this is weird things. I think from the mathematics. But of course this is the identity is exact. So I believe there are some mathematics in our brain states, but people try to figure out how to be realized in the connection into our brain states here. So now we have the order parameter, right? Because we have the different kind of brain state, and the brain state can be had to correlate with each other, and then we can have how correlation metric, the QAB. The AB now is a metric because sum up all the neural index, and then you can go to the correlation between different kinds of state and this one. And very weird, from this is the reason we have the other parameter is called RAB. RAB it means that it has a mathematically the conjugate in real parameter. This is S hat, but this feed is a real activity from the brain state B, but this is from brain state A. Then what do you mean by this term? Then we do some calculation, use some mathematical transformation. We know now this is R. Why did they appear as a hat here? It's just due to a derivative. You do the derivative with the mean population of the activity with some external current, and then you can get this term. So if you use this is a typical mathematical identity, you know the R it is a response activity. Response parameter of the neural circuit. So now because you can inject some external current into your neural pool, and then to see how the mean population activity elaborate. That is, you can test the sensitivity of the neural population, and then it means that this activity will come out from its response activity. So it means that then we have two parameters. One is the activity correlation or brain state correlation, and the other it is a brain state sensitivity. So it means that these two kinds of the metric it complete the story about this non-equilibrium fixed point. So now people want to has some the probability test on this kind of experiment. So now we look at a very beautiful result. So if we look at the Q, right, and then you can draw the curve. We have the two free parameter. It is G, it is the strength of the coupling, and the gamma, it is a correlation between bidirection of the coupling. If you tune these two parameters, you can see has a critical line, and beyond the critical line, it continuously changes to a non-positive value, and this is 0 value. Of course, we know 0 value it is fixed point, trivial fixed point, and the positive value, of course, is chaos. So it means that we now can identify this as a continuous transition because they can destabilize the trivial fixed point, and then you appear the chaos. So we can use this order parameter to characterize this, but before in 1988, people do not know this kind of order parameter. You just use some correlation or something like that. And this is Q, and Q is a diagonal or non-diagonal. If you look at the response, it's quite out of expectation. If you look at the other response, you mean that now you look at this picture, it has a critical line, it has a peak. right? Has a peak, but this peak is asymmetry, right? In the chaotic side, it decays slowly, but in the non-chaotic side, it increases rapidly. So, but the peak is always at the edge of the chaos. So this, I think, is the edge of chaos. It means that people always doing some reservoir computing on this is the critical line. They do not doing about this deep chaotic or do not doing about tree of this point, because if you look at the response, because on this case, on this both side, the neural network doesn't respond to the sensory input. Of course, it cannot do memory, cannot do calculation. But on the critical line, everything it is a peak. It's very sensitive to the sensory input. So we found a mathematical evidence about this is related to the memory of the system. Memory capacity is it take a minimal. But very intriguing things, because in the PNAS 2022, because doing some human data recording, and they found some on the edge of the chaos. There has a peak on the information richness. That is, so you can measure the time sequence from the different kind of CG sweep and waking. And they also find a maximal point here. But here we use a Toya model. We also find a maximal point here. I think these two things, things happen on the different contexts, but it things has a unique mathematical origin. It make us to very exciting. We can find establish some mathematical structure, use a response parameter to calculate some human data, the fMRI data or something like that. And then you can look at another things that happened in 2024. It's very interesting. It is use brain complexity. People use the perturbation complexity index, and then this PCI is used in a clinical application to determine the conscious level, right? So if you use the population size to increase, and then this PCI, and then you will be peak around this is a critical line. So it means that now in the hospital, people use this PCI to identify the conscious level. Here, now we can ask a further question, whether PCI related to our response parameter. So this, I think, different kind of thing. It seems at this level can be connected together. Even you can address some hard problem of the consciousness. Because consciousness, you need a responsiveness to the external perturbations. So now I skip here, and then we just to look at the classical paper 1988. That use the dynamical mean field theory, and then you can use in the dynamical mean field. If you focus the correlation function, you also find the continuous transition. If you use the integral response, you can also find the peak. So it means that in this sense, our theory is very surprisingly consistent with the previous old theory. So now it means that we can create the second type of the tool to understanding the dynamical system, the dynamical of the neural dynamics. So now this auto parameter, and also a very very interesting thing is if you zoom in, this is increase of the response parameter, and you will find, and this is a rapidly increase, and then the slowly decay of the response parameter, and the rapidly increase, it means that actually in complex system, this is a tipping point quotient. If increase, it means that the system now is very close to self-organization criticality. There will be some chaos transition, but you know on this side there's no chaos yet. But if you increase the synapse gain, it already has some signature about the chaos transition. So this is called the tipping point quotient. We can doing some tipping point quotient in the data analysis of the emergent behavior of the brain dynamics. So, of course, this is very crazy because they can connect to the psychedelia, because the response parameter may be there. Because the response, it makes a correlation between this state and the other state. If you use a psychedelia, because they can reshape the geometry of dynamic landscape, this is called the psychedelia Huang hole. This is crazy concept. But here, after you take the drug, and then you can move from one attractor to the other attractor. I think this may be intimately related to the response parameter calculated in our theory, but this is to be verified in a further experiment in the psychedelics. So now, because the time is limit, I just use the field limit to tell about how useful our theory to doing the dynamical system. The one is a freezing chaos, right? The chaos now can be freeze because before, you know, the stimulus or noise can be freeze chaos, and also in recently by Larry Abel paper, you can use the Hebbian plasticity can be freeze chaos. But from our theory, from this paper in the 2025, we invent the third way to freeze the chaos without any synapse plasticity, without any stimulus, external stimulus. Then let's look at how we can do this. I'm sorry, this is a Chinese character. This is a neurodynamics control, and this is working memory, and this is imagination. And this we know this on-cycle feedback term, right? This feedback term is very important. For example, previously this is a chaos. If without this term, and the dynamics is chaos, very high state. But at this moment, you add this term into your dynamics, and then the chaos will become stationary, and the chaos will disappear, and then it will be stable representation of the neural activity. And then during the period, you can doing some working memory or something like this. This we call the persistent maybe brain state. And after some moment, an arbitrary moment, and then you just shut down this term, and then they come to the chaos again, right? So then this on-cycle feedback term can be connected to region in the phase space, fully depend on when you want to control this system. So if you look at the, sorry, I just come to here. If you look at the low dimensional projection, and then I think originally you're starting from here, and then this is origin curve, it is a chaotic region, right? Then up to this point, and then you just to turn on the feedback term, and the dynamics just come to the non-equilibrium fixed point, because the blue term, it is non-equilibrium fixed point. If you look at the kinetic energy, this is a transition dynamics, and the kinetic energy decreases sharply, and then they reach a fourth stationary region. This we call the non-equilibrium steady state. This we call the chaos. Chaos is here, right? And then if you do not switch on the feedback term, and the chaos will be maintained a very long time. But now up to some point, you switch on, you turn on the feedback term, and then the kinetic energy decrease further, because you want to reach the fixed point. Fixed point has a zero kinetic energy here, right? So then you will go to the zero speed, right? So this is non-equilibrium fixed point, non-equilibrium steady state, and the transient dynamics. So in any kind of high-dimensional neurodynamics, that is ODE, you have the three stage, right? So now, using our current theory, we can control. This is from non-equilibrium steady state to non-equilibrium fixed point. We can build a bridge, and this bridge in the previous theory, no bridge, because the fixed point is strongly unstable. So now we can control the trajectory flow in the entire phase space. So we can use this to do some computation. For example, some prediction, some memory is so very compatible performance with the standard recurrent neural network. Though gamma equal to 0, it is a standard the neural network, but gamma equal to 1, it is our control neural dynamics. It is very to do some reservoir computing, something like this. And the last point, as mentioned, this will come to this part. We can lead to the new algorithm and hypothesis to this parody. Now we can look at we can use this kind of the lens one dynamics as a generative model. If you look at, because in the biological sense, we should make every computation should be local, and has asymmetric plasticity. The Haiping, it is just an abstract of Toya type of the brain. But we also have a state transition in our brain, or from one state to the other state. So this is three necessary components. It's very important for a biological plausible generative model. But if you look at the generative model used in machine learning, all fail in these three criteria. For example, restrict Boltzmann machine provided by Hinton fails, and the GAN provided by Goodfellow fails, and VAE provided by some guys, very famous guys fails. But even now, the generated diffusion model also fails because it has a score function. You cannot imagine how the brain implement the score function and the feed-forward, back-forward dynamics. It's too complex. But now we use this our theory, we can create, because we have a kinetic energy. You think about in our brain has a kinetic energy potential, and then we had a fixed point. Now the fixed point is non-equilibrium fixed point, and then this non-equilibrium fixed point, we already know their distribution. Then we can use this distribution to store information from the sensory inputs, right? So now we're just doing this. Now we have a new kind of the brain dynamics. Use this brain dynamics, right? But we have the kinetic energy, and then it has a very nice distribution. And they have these three components. So we can design a generative model to fit any kind of the sensory input, right? You just do the Carroll divergency. This P, it is now our kinetic energy distribution. And this data is any kind of the sensory data, right? And then you just to calculate this Carroll divergency, because we already know the P, right? We do not know the P data, but we know the P. Then you can insert the form of the P, and then you can do calculation. This is quite easy, and then you can finally get out. Then you can minimize the Carroll divergency. Is equivalent to minimize two term, because this is dependent on data, doesn't depend on coupling. So we just to remove it out. We don't care about this first term. And then the second term, the log Z is the free energy, right? We talk about, because dependent on the coupling, right? And this energy term, of course, kinetic energy dependent on the coupling, right? So this two term is the coupling dependence. So then we can minimize this two term, and then to optimize the coupling. Because we optimize the coupling in our brain science, we call the plasticity, right? So then we can devise some plasticity rule and to learn, for example, this natural imaging, and also this is the handwritten digits. And then we can look at very simple. You just do gradient with the recurrent coupling and learn this recurrent coupling. But finally, you will find the plasticity is local, right? You have the two term. One term is dependent on the data, and the other term is dependent on your model. The model, it can be generalized by our Brownian or Langevin dynamics. If you look at this term, it is quite surprising, because now this is SK minus HK. SK minus HK just the neural speed. It is speed, right? And this phi SL is the neural activity, right? So the neural activity coupled with the speed to create a new type of plasticity. I think up to now, no one talk about the plasticity can be incorporated, how fast of the neural changes. Maybe how fast of the neighboring neural changes will affect the plasticity, and then it can give you a Brownian dynamics has a symmetry learning rules, and then we can see the results. Then after learning, and then you just to use the random initial connection from the phase space. This is a PCA projection, and then you start from here, right? This is a green part. Green part is the digit 1, and then the brain activity is the digit 1, and then you just use the white noise to drive the dynamics, and then you will become to the digit 1. That is a broad digit 1, and then they'll come into the digit 6. And then finally they'll come to the digit 3. So it means that they can realize the brain state transition in the phase in the phase space, and this is driven by the local plasticity rule. From here, if you retrieve each trajectory, you can see the trajectory is highly noise. But if you put all the activity into a whole picture, you can see this is noisy trajectory can give you the mental image. For example, this 3, 1, 1, 6, and then if you continue, then they can switch different kinds of the generated picture. And this 3, it is different from your training data. This is creative by the neural circuit. So now we can use this simple principle. We can create a new kind of generative model just based on the brain circuit dynamics, and use a local computation, state transition, and asymmetric plasticity. So now I skip here, and we can talk about some take-home message. Now we have energy minimization is a powerful principle for us to consider neurodynamics, and then why different mathematical tools lead to the same result, because we have the different way to control the chaos. But here we can have another way without plasticity, without stimulus inputs. And also the new principle framework, for example, maybe start from here, we can invent some new principle framework. Maybe this is a future work. to unify the diffuser and transformer, maybe we can use this framework to do generative process, even we can do reasoning and can do imagination and whether you whatever you want to do here. So I stop here.
Thanks for Louis. Actually, the title is, yes, it's bridging the network and circuit model. So I already noticed that actually many of the interesting topics discussed in this summer school relate to higher level cognition. And you can always train a fancy neural network model to mimic the behavior and the behavior of some cognitive functions. But I also know that a lot of labs here, they focus on the microscopic things in biology. For example, the connectome and all these different cell types and the different physiological properties exhibited by this microscopic circuit and the neurons. So that's in this advanced lecture, I will discuss a very simple, actually, mathematical framework that can link the network level model and the circuit level model. So this is a work can be traced back to my postdoc with Sergei Nostoyek and in collaboration with Eric Shebran, David, and Stefano. It's actually, it links the two lines of the work in Sergei's group, actually. One is the low-rank framework developed by Francesca and also can also apply to machine learning field by Friedrich. And another line of work is one of the surgeon's postdoc, Daniel Martin, and also a talented mathematician, Hu Yu. Yeah, I think he will give the talk in another summer school after our CNeuro, but if you are interested in mathematical things, then you'd better to go to Westlake University and then to listen to Hu Yu's talk. So actually, I'm the people from the community who care about neural network and neuron population, and we always consider that the computation and cognitive function that can is emerged from a population of neuron or large neural network. It's not. I'm not claiming that the single neuron doesn't matter, but usually we are the people considering a population of neuron as a whole, and which can execute, which can implement some specific neuron computation and cognitive function. But there is another group of people who want to really use this idea of biologically inspired network model, like the circuit model, and they really care about the single neuron level and even some compartment within a single neuron. So this kind of that community, they focus more on the circuit level things. And for the circuit level, we always consider these different cell types. So the simplest way to classify different computational units is to classify them according to different cell types. And usually we will use the average activity of a specific cell type of population of neuron to describe the activity of this circuit. And then regarding the connectivity, Then as I said in my first class, we, apart from the unit, computational unit, we also care about connectivity, that is, edge between different units. And here, because we just simplify the activity of cell type as an average activity, so we only consider the average connectivity between different cell types as the synaptic coupling or the network circuit architecture. But for the network version, we have heard many talks about these task variables and computations and this mixed selectivity mentioned by Albert and the distributed representation and also the low-rank dynamics, not the low-rank, low-dimensional dynamics that can correlate it with some specific task variables, okay? And corresponding, like, relate to these two different community, network community and circuit community, we have two different type of model, right? We have these recurrent neural networks, densely discussed in this summer school, and we can train the neural network and then extract the low-dimensional dynamics, and then we relate the latent variable to this kind of abstractive computation and the representation, right? And another side, we have this circuit here. We have different neurons, like what mentioned by Albert, this Wilson-Cowan model as the classic and the queen of this field. And also the course by Adrian, she mentioned this kind of specific neuron circuit underlying the bird song, right? And you can see this is another type of model we can have. Then the question is, how can we link these two models? Can we find some unified theory or some unified framework underlying these two look and similar neural network model? I just want to mention that if you think about this circuit model, actually it's still low rank or low dimensional, because you take the average of the population, you reduce the dimensionality or the complexity of this model, so it's by nature low dimensional. And the dimension is actually depends on the average, the cell types and the computational units you have in here. So to link these two different type of model, we will focus on these questions. First is, how can we reduce a large neural network to a low-dimensional model, like simple circuit type model? And second is, how do the low-dimensional units correspond to the average cell type activity? Also known as, for example, whether the low-dimensional, each dimension correspond to one cell type, or each dimension correspond to a linear combination of like different cell types. And how does average cell type connectivity correctly predict dynamics? When can they correctly predict dynamics? Are there any limitations? And the fourth is, how can the heterogeneity, for example, across different neuron population, can interact with the low-dimensional dynamics we mentioned or were usually found in the recurrent neural network? This kind of linking is not a new question, actually. It's not a new thing. Many people have put their endeavor into exploring this link between these two different type of models. For example, if you know a series of work regarding the Wilson-Cowan model, you will know that they have a lot of study using this local connectivity statistic and the dynamical mean field theory to probe the network dynamics in EI type neural network. And also a lot of work currently using this low rank framework and to study the global connectivity statistic and how this global connectivity statistic give rise to the global low-rank dynamics, low-dimensional dynamics. And what I did is, based on this classical and traditional work, and do some combination, and then have a one step further towards linking these two modeling. And I don't want to give this background anymore, because you can always find very excellent explanation from Adrian's talk. And also, I have already given you some background in random recurrent neural network and low-rank recurrent neural network. So I will directly go to my result and the introduction to this part of advanced course. So I will, in detail, discuss the local connectivity and then to the global connectivity. So for the local, I start from the local connectivity, which is the EI network. And when you try to define the statistic of this local connectivity pattern, you will consider to describe the statistic of the connectivity strength. And so in the Gaussian random neural network, I told you, I discussed in the first lecture, we just used the Gaussian distribution to describe the variable that is Jij, right? And then if you consider in the if you are doing some connectome data set and you are recording this kind of connectivity, it's very hard to find the exact physical variable that is exactly the JIJ, the Gaussian random variable, right? And how you somehow find the mapping between this experimental data set with the quantity we defined in the model. So then there is an answer. That is, we use the probability of a specific a type of connectivity between two different neuron populations and use the probability as the representation of the connectivity strength, that is JIJ, which means if these two populations have a denser connectivity probability, then the synaptic strength we use in the model can be larger. If it's sparser, then the connectivity we use in the model can be weaker. Right? And then this is actually first order statistic. So you can find that actually there is only one synaptic coupling exists in this cartoon here. So these describe the first order statistic of connection strength. And as I said, this kind of local connectivity has been long used in study the EI network dynamics. For example, this kind of work from, this EI network from Sompolinsky and M.H. Brunel. And then you can use some mean field theory to reduce the complexity of this random neural network and to get this effective EI circuit. That is actually what I mean by circuit type model. And then once you have this effective EI circuit, you can get the low-dimensional description of the dynamics from this circuit model. And you can, as I said before, you can use average cell type activity to represent the dynamics of units, each computational unit, and then you can describe the connectivity between different computational units using this average cell type connectivity. But when do this kind of reduction, you need to have some sort of assumptions. Two important assumptions we have before doing this kind of reduction is, one, the cell type dependence. So this kind of averaging is averaging within each population. So both the computational dynamics, the computational units, and the connectivity depends on the cell type. So there is a cell type dependence. Another very important assumption is no correlation. So the so-called identical independent distribution. So which means, for example, the JIJ is independent from JJI, and the JIJ is independent from JJK. And so there is no correlation allowed in this kind of reduction. The spatial correlation is so here I didn't mention the spatial network. So there's no dependence on spatial things. So I understand if you have this spatial arrangement, then the connectivities depend on the distance, and that is kind of spatial, but that is beyond the scope of what I discussed here. Yeah. Okay. As I said, how can we get this local connectivity statistic from experiments? So this is a very beautiful work by Allen Institute. They have this science paper 2022. They use this in vitro patching to study this connectivity probability in both mouse and human brain in visual cortex. And they have this connectivity probability. And as I said, if you believe that the connectivity probability can be precisely matched to the connectivity strengths across neuron population, and then you can directly use this probability in this color map to represent the average connectivity used in your model. But what I want to say is, in their data, not only the first order statistics, this connectivity probability can be precisely found. They also found something beyond the mean connectivity. That is the correlation between synaptic pairs. So we call this connectivity motif. This is just a name. You can call what type of name you want to define this kind of things. So there are 4 typical types of synaptic motif, second order synaptic motif: the reciprocal motif, the chain motif, divergent motif, and the convergent motif. So the reciprocal, you can consider it as like symmetric connectivity, which means the connectivity from I to J is correlated with connectivity J to I. Of course, it can be totally synaptic, symmetry, or like just partially symmetry. It depends on some kind of parameters here. That is the tau here. Represent mathematically, it's the correlation coefficient beyond, in front of this kind of motif. And this is what I said they found it also in the experimental data. So they found this kind of motif, not only in different type of motif, but motif across different neuron populations. So you can have the EE type of reciprocal motif, and you can also have EI type of reciprocal motif. That is what I mean by like you can have motif from different type as well as different neuron population. So these are what I want to discuss in this course about local connectivity, and then the global connectivity. The global connectivity, I will use this low-rank framework, of course. And this low-rank framework, so this is the most basic low-rank framework. You have this random connectivity. You can decompose it into the low rank, and you truncate it R to R. And then there is also a correlation, right? I said that the correlation between M and N significantly determine the dynamics of this low-rank framework can have, right? So through this using under this low-rank framework, the effective circuit here, the connectivity between different effective circuits, as what Professor Bing Ming have mentioned, different factors are determined by the correlation between M and N, right, after some nonlinearity. So this is another type of model, and the dynamics of this model also is controlled by the correlation to some extent. Then I can have these two models together. So for the local connectivity, we have this population, and we have average activity. But when we do not quantify the higher order moment or higher order motif, we do not have correlation here, just first order moment, right, the mean connectivity. And for the global connectivity, we do have the concept of population, but it's a computational population, it's a functional population, it's not a real population we define in biology. But this kind of model, it directly has this correlation, the concept of correlation. So what I did is to somehow combine these two approaches. So the computational pipeline I did in this work is I start from defining a network from local connectivity based on multiple cell types. So I do not assume a homogeneous neuron population and then train it, or just use some global statistic to describe this. I directly develop a network, I directly start from this local connectivity based on multiple cell types, and then I approximate it by the low-rank network. And therefore, I can reduce the network to low-dimensional model and can describe the dynamic and interpret and can have a clear interpretation for the dynamics that can host by this low-rank network emerge from the local connectivity. And I will tell you later. Yeah. And finally, I will compare the dynamics exhibited by this kind of low-rank approximation network with the one based on the classic mean field reduction, that kind of mean connectivity. So the approach, as Rava asked, how do you do this low-rank approximation? I will do it step by step here. So currently there's no mean connectivity for neuron population, but I just want to give you some mathematical derivations behind this kind of approach. So we start from the connectivity, random connectivity, and we do the matrix factorization. Notice that here in this work, we use eigen decomposition, not SVD. So there is some small differences, but it doesn't matter. Just keep in mind that I use eigen decomposition. And then I keep the first R rank here. And then I just use the first R rank to and regard it as the low rank approximation for the original matrix. There's no population here. But I will tell you, I can do the same thing for the low rank, for the multiple neuron population network. So currently we have different, still a random matrix here, but different color represent different connectivity block with different pre and post-synaptic neuron population, right? And in this network, I have both first order statistic, that is the mean part, as well as the second order, the motif part, the correlated part. And any type of this network, random network with mean connectivity, can be decomposed into the mean part and the fluctuated part. So the mean part corresponds to the mean connectivity, which is determined by the first order statistic here. And then you can see that there is a clear structure in this mean connectivity. So the mean connectivity is a block matrix. So this block matrix should be a low rank matrix. And the rank, if each element, each constant element in this mean connectivity is independent with each other, so they are heterogeneous, and the heterogeneity, then this block matrix is low rank, and the rank is the rank depends on the number of populations. For example, here are 4 different neuron populations, so the rank of this block matrix is 4. And for the fluctuation, I will start from the, because the mean part is already in this part, so this part has zero mean, only the fluctuated component here. And I would tell you that I can analytically calculate eigenvalue of this random matrix, which can be decomposed into mean and variance. So I will start from the method part that I used. I actually used the eigenspectrum analysis and eigenmode decomposition. So we start to think about the simplest version. We have a unique rank, whatever. You can think about it as this mean connectivity or whatever kind of unique rank you can have. And then you have a summation of the unique rank structure with a totally uncorrelated random component. And eigenvalue spectrum look like this. There is an eigenvalue bank, which is determined by the random component, and there is an outlier, which corresponds to the unit rank. And the value of this outlier, if you know any type of eigenvalue decomposition, you know that this value is exactly the inner product of M and N. Okay, so if this random part is totally random, there's no correlation, then the value is exactly the M and N, the inner product. But if you have any correlation within this randomness, things change. Then the randomness, the correlated random component in this random matrix might affect the eigenvalue here, the eigenvalue outlier here, which means there is some interaction between the unit rank with the correlated random component. Of course, you separate these two, but they have some nonlinear interaction within each other. And this is not the whole story. You can even have a multiple outlier emerge because of the perturbation caused by this correlated random component. And this mathematical derivation you can find in this paper by Friedrich. And if you have this random component, and then if it's correlated, and then it causes emergence of two eigenvalue outliers, then you can no longer use just rank 1 model to do this approximation. Then you need to use the rank 2 model, which corresponds to the 2 eigenmodes determined by these 2 outliers. Okay.
What's the meaning of the random correlation here, and it will not affect the bulk between them?
So if you only have this unit rank, the eigenspectrum looks like an isolated point here. That is outlier. The existence of the bank is only because of the existence of the randomness, because each point corresponds to the 1 rank here, right? The correlated means, like, for example, symmetric and or the reciprocal or the chain motif. Good question. So there is another work from Terrace Tao, who is a small brother of Tao. Yeah, so what they did is they used the low rank part as the perturbation to the random part, and they explored how the low rank part will affect the bank, the eigenvalue bank. But what I did here is how I used the random part as the perturbation instead of the low rank part as the perturbation. So I'm mostly focused on how the perturbation, how the structure in the random part can affect outlier. I'm not doing the reverse one. Okay. So all these are math. But why the experimentalists need to consider this, right? I will give you some example. I showed this figure in Hong Kong, and Quan argued me, but I still didn't have the time to find the answer. So this is the connectome of. Sorry, this is the brain of the Drosophila. And if you do the eigen decomposition and try to find the eigenspectrum of this Drosophila brain, and you will find that actually there are many outliers. It's not just a random matrix here. And if you look at the connectome of this zebrafish, and you can also find this outlier here. Okay. So it does matter. And then I will tell you, so I haven't mentioned the multiple neuron population or the EI cell types yet, so I will discuss it now. So as I said, I did a similar thing as what I showed you before. I decomposed this this random connectivity matrix into two parts. One is the mean and another is fluctuation. Sorry. Regarding the mean, I used the Brunel type model, so which means I didn't, the mean connectivity only depends on the presynaptic neuron population, so it doesn't depend on the postsynaptic neuron population. And I have the fluctuation here. And then, as I said, the block-like connectivity structure is actually rank 1. You can do this very simple linear algebra, and you can have this unit rank structure plus and random connectivity. And also, as I said, if you have uncorrelated randomness, and you will no longer see, there is no impact of this random component on the eigenvalue outlier. So the eigenvalue outlier is exactly determined by the mean connectivity. In this case, you can still use the rank 1 approximation that is exactly a mean connectivity plus some random IID randomness onto this mean connectivity. So it doesn't change the response, it doesn't change the global dynamics of this network. As what I showed you before, like if you have uncorrelated randomness, and then the rank 1 approximation does the same as a mean connectivity. How about reciprocal motif, which means there are some symmetry in the random component? We did the same analysis, but instead of uncorrelated randomness, we have this correlated randomness, and we found that actually there is a very small shifting of this eigenvalue outlier, like push the eigenvalue outlier to a more negative part. And we can do some fancy math, and we can analytically predict this kind of deviation caused by the reciprocal motifs in the random component. That's why I want, so here is a point I want to mention, work by Professor Yu. So you can already see that actually this the correlated randomness not only caused the shift of this eigenvalue outlier, but it also caused a larger eigenvalue bank. So this is another effect can be caused by the correlated randomness. Yeah, but as I said, if we think that this eigenvalue outlier and the dominant eigenmode dominate the global network dynamics, then we can still ignore the random part correspond to the eigenvalue bank and just use rank 1 model to approximate or to approximate this full rank random matrix and to do the further prediction of the network dynamics. But still, this kind of deviation or shifting of the eigenvalue outlier is really small. So we want to add, so we discussed with our collaborator, David, Stefano, and Eric, saying that, oh, so does the motif really matters? Maybe the motif is not as important as we assume or we claim. And then Eric actually told us, no, wait for a minute. We do see this reciprocal motif, the absolute value of this reciprocal motif, the correlation coefficient in front of it is really large compared to others. But when we do the simulation, when we construct a network with this specific type of motif, we found that these guys, the chain motif, which means you have a long chain, you have information from I propagate to J, and to J propagate to K, and to K propagate to G. That might lead to a very large deviation of the eigenvalue spectrum. So we start to think about whether we could develop a framework that can explain the impact of this chain type motif. So with that problem in our mind, same figure, but now the correlation is not caused by this symmetric connectivity or symmetric second order motif. Instead, it's caused by this chain type, like this feed-forward propagative-like motif. And after develop or incorporate chain type of correlation in the random component, we found similarly the eigenvalue outlier. So we use, I should mention earlier, so we use a mean connectivity with inhibition dominant. The eigenvalue outlier, the mean originally determined by the mean connectivity, still is pushed leftward. But we found very interesting is that there is some positive eigenvalue outlier emerge from the eigenvalue bank. But remember that the mean connectivity is negative, is inhibition dominant. So the only reason for this weirdo is the chain motif. Yeah, so the JI is stronger than JE. So the lambda 0 is less than 0. So if you just consider the mean connectivity, there's by no chance the network can be activated because it's inhibition stabilized. Okay, and then if we consider the actual eigenmode correspond to this newly emerged eigenvalue outlier that is positive, then we can no longer approximate the original random matrix as the ranked 1 model. Instead, we need to use the ranked 2 model as the approximation. So at this time point, we need to approximate the original connectivity as a rank 2 matrix. And I will show you that we can also have the very similar theory. We can calculate, we can find that actually the eigenvalue outlier does increase with this, the chain motif, strength of chain motif, and we also found other parameters also matters. So for example, the size of the network. Yeah. Here is the only math slides I have here, actually. There is somebody maybe focus on or care about the regularization, right? So there are two different regimes. So as I said, if you only use the sparsity or connectivity probability to describe the network connectivity, and then you should be under this regime. And otherwise, you have this strongly connected regime, and then it might require different balance condition for this network. But it's another story. I will not elaborate on this. And then, after having this mathematical framework, we really want to link our work to biology. We want to link, or we want to tell us the complete story, saying that, okay, you have this framework and you have this low rank approximation, and how does this give you beyond the classical mean field theory or classical neuron circuit model? And then we found a very interesting phenomenon called paradoxical effect. So paradoxical effect, what does it mean? So as Albert had showed the example, like when we want to probe a response dynamics or response properties of specific population of neuron, and then we give this optogenetic perturbation or any type of external input to this neuron population, and we see how the response of this specific neuron population we are interested in change, right? And usually if you think a normal condition, if you inject, for example, you inject input to inhibitory neuron population, and the activity of that inhibitory neuron population should increase. But some of the study they show different thing. So especially the work from Ken Miller and Agustina and Alexander Sanzani, they show that a wide range of brain cortex, the dynamics reside in the regime called inhibition stabilized network regime. And in that regime, one interesting phenomenon held by that regime is this paradoxical effect, which means you give this external input to inhibitory neuron population, instead of increase the activity of that neuron population, the activity of that neuron population decrease. So it seems like a paradoxical. You force me to increase, I do not want to increase. That is what I mean by paradoxical effect. And as Albert mentioned, that in the classical mean field theory, the cause of this inhibition, cause of this paradoxical effect is because of the strong inhibition in the network. Without that strong inhibition, the excitatory neuron itself can excite itself. So there is a positive feedback by excitatory neuron population itself, and that caused the network to be unstable after removing the inhibition neuron population. So only when you add a strong inhibition, and that inhibition population can stabilize the network, if you do some phase plan analysis, you will find that the inhibitory population exactly hosts this kind of paradoxical effect. But what we found here is, so we want to use our framework to see whether the random component beyond the mean part, beyond the mean WEI, WII, WIE, and WI can give another direction to regulate this kind of response function and pushing this dynamical regime, for example, from non-paradoxical to paradoxical or vice versa. And before doing this kind of simulation, we still want to see whether there are some mathematical work we can do to, like, approximate this kind of, to guide us to do this simulation. So we calculate the linear dynamics of this network, and then we find the fixed point of this ODE system, and to find the ODE system, you need to calculate inverse of this random matrix, which is n by n. If you just have this n by n matrix, you want to calculate the inverse of an n by n random matrix, really hard. But because we have this low rank approximation, so we can somehow skip this kind of complicated computation of this random matrix, inverse of a random matrix, and the final equation for this fixed point is very simple, so it can be decomposed into the contribution from the external input, and then there is a decomposition of the contribution from different rank, this rank 1 and rank 2. Very simple. And also, our previous calculation of the eigenvalue outlier is not like useless. We can still use the precise calculation of the eigenvalue outlier to probe it into here and help us to develop a more accurate approximation of the response function or the population neuron response. Okay. And when no questions? Okay. So we start from analyzing how the neuron population response change with the change of the correlation coefficient, or the change of the abundance of the chain type motif. And we found that when we gradually increase the chain motif strength, tau C, and the population response of excitatory neuron population start to increase, which means the network dynamic is activated after like a stronger and stronger chain type motif. But I should mention that I only increased the correlation coefficient. I didn't change the mean, which means if you only think about or you only consider the mean connectivity, the response should look like this flat line. So there's no change in the network activation. And this is not a powerful part of our framework. So beyond like predict this neural network activation, we can also decompose the activity to the contribution from different rank, as I show you in the mathematical equation. So, tau C? Tau C is the correlation coefficient between Jij and JJK.
But so these results are not for the low rank? I thought these were for the low rank.
Yeah, this is for the low rank.
But you add, so it's just this low rank plus some perturbation which has that correlation?
So we have the mean that is low rank plus a perturbation, the random component, and that the sum of these two parts can be approximated by another low rank.
And the reason that it's simpler than for the paradoxical effect, the reason it's simpler is because you do something like the Sherman-Morrison.
Yeah.
That's what.
Yes. 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 Should I explain Sherman-Morrison? So it's a way to compute the inverse of a large matrix. So if the large matrix, the J can be decomposed into some low rank factorization, then you can use the Sherman-Morrison equation. And then instead of calculate the inverse of this n by n matrix, you just need to calculate the inverse of the R by R matrix. Yeah. Okay, we can also give you show you that actually the network activation is not caused by this eigenvalue, which is determined by the mean component, but it is determined by the emergent eigenvalue outlier that is caused by the chain motif. So this one. So this one is just negative, so it will stabilize the network forever. But once you have this emergent eigenvalue outlier, the network dynamic will be activated. So this is one thing we found. And how about the paradoxical effect? So the paradoxical effect, as I said, has been tested in experimental experiments using like this optogenetic perturbation. If, for example, usually we perturb the inhibitory neural population, and we found that there is a damp because of this paradoxical effect. And then we did a similar thing. So the paradoxical effect means you give this external input to the eye population, and then you find the response of the eye population decrease. So we focus on this chi-II, which means it's actually a derivative of firing rate of eye population over the input given to the eye population. And we found that there is a crossing point. I still didn't change the mean connectivity. The mean connectivity is still the same. I only changed the chain motif strength, like from 0, which means there is no chain motif. The randomness is just random, IID. And then I gradually increased to just a very small number that is 0, 1. And I found there's a cross point represent a change from non-paradoxical to paradoxical. Remember, I didn't change the mean, which means when there's no chain motif, the mean connectivity keep the network in a non-paradoxical regime, which means the mean connectivity is not inhibition stabilized the network. But instead of change the inhibition, mean inhibition, I change the chain motif, and which push the network to undergo this paradoxical response. This is not a whole story. The whole story is we still want to make the framework more biologically plausible. So instead of considering the Gaussian network, the network with Gaussian synapse, we consider the sparse network, which means the variance and the mean connectivity or the connectivity probability, they are correlated. And also, because you have the sparse network, you can no longer define by hand the sign, or whether the correlation is positive or negative, because if you have, for example, the tau EI, which means the first synaptic inhibition and the second synaptic sparse and excitatory, and then the tau EI should be negative, and the tau II should be positive, because you have the first inhibition, and the second is still an inhibition, the chain. I found very interesting thing is that there seems to be a paradoxical effect on the paradoxical effect between Gaussian network and the sparse network. I did the same thing. So the absolute value of the chain motif strength here is the same across different neuron population. That is tau EE equals tau. The absolute value of tau EE, tau PQ, they are all identical. But instead of the anticipated change, like alternation between non-paradoxical to paradoxical for the sparse network, we no longer see this paradoxical transition. So we start to think that maybe the EI structure, the Dell's law, does matter. So we did a complete check of this parameter regime. For example, we checked different negative tau EI, IE, which correspond to the sparse case, and also positive tau EI, IE, which is mostly artificial and correspond to the Gaussian setting. And we found we reproduce, or we can predict very clear transition. For the sparse case, there is no transition from positive to negative. It keeps in positive regime, which means there's no paradoxical effect emerge. But for the Gaussian case, which means you fine into the sign or the positive or negative of the tau of different heterogeneous chain motif, and you can observe this kind of paradoxical effect emerge. So what this give us? So this give us that first, these higher order moment in the connectome data set really matter. Maybe in some case you can always fit mean connectivity and you can capture most of the dynamics, but there are certain sort of connectivity dynamics cannot be captured by the simple mean connectivity. And maybe it's good way or a good strategy to think about higher order connectivity circuit. And another thing is, it's simple to consider a homogeneous network structure. But if you include the heterogeneous property, no matter across different local connectivity circuits, nor across different neuron populations, it will complicate your analysis, but it will also give you more richer repertoire of dynamic you can find. I don't know whether I have more slides. Oh, this is the last slide. Okay. So that was the version I gave in Italy. And after that, so I present this work to Agostina, and then Agostina argued that because in their general framework of paradoxical effect, they show that they have a framework, they have a theory saying that, as I told you, if you want to check whether a neuron population have this paradoxical effect, you'd better to remove that neuron population. And if the remaining part of the connectivity, the dynamic of that emerge from that remaining part is unstable, then the part you remove should have paradoxical effect. And actually, we didn't find that because, we cannot directly remove the inhibitory neuron population from our framework because you have this second order motif. So you have these higher order things. If you remove that part, you destroy the higher order correlation. So we try to solve this kind of a match between their theory with us. So we developed this effective connectivity. The intuition is very simple. So we think that second order matter, but in our original network construction, we do not have this correlation because it's random. Only the second order can keep this kind of correlations. So we, instead of use this type of decomposition or construction, we develop this effective connectivity that can incorporate this second order statistic through an averaging across Z square. So we can include the correlation here. And after do this kind of effective, once we have this effective circuit connectivity, we can calculate the not eigenvalue spectrum. We can calculate the response function or the inverse of this matrix, and then we found that, oh, Rava, this is what Sherry-Morrison. Okay, it's another name of Sherry-Morrison. It's just to calculate the inverse of a matrix. And then you will end in the same expression. And in that way, we can, actually once we have this, we find the exactly precise match with the theory proposed by Miller and Agostino is saying that once we remove for example, the inhibitor population from this J effective connectivity, if the excitatory part or the remaining part is unstable, then the inhibitory neuron population has this paradoxical effect. Yeah. No. So it depends on, for example, if you just one E and one I, then JEE, if JEE is less than 1, then it's still stable because there's a leaky term. Right? Okay. So we did the thing, so we replicate what suggested by Agostino and Ken Miller, like we find this bifurcation or this boundary here using the effective network. So when the eigenvalue of the effective is 1, as I told you in my first class, then the network has this stable and unstable transition. And you can see that actually at this time point, at this line here, the response function is exactly 0, which means there is a transition from paradoxical to non-paradoxical. So we finally managed to link the two genera. We claim it's a general theory between us and theirs. So the last slides I have, I answered this question maybe. So when and how can we reduce a large network to a low-dimensional model? So it can be computational origin, for example, what Professor Bing Ming and also my postdoc supervisor Ostoik did in this development of low-rank framework. And also it can also be the cell type origin. For example, you have this block-like structure. And when do low-dimensional factor correspond to average cell types? When they correspond to low-rank structure, and then this low-dimensional can directly link to the cell type. And when does average cell type connectivity correctly predict dynamics? We say that maybe the strong deviation caused by this motif is not so important, or it's not caused some qualitative change, then it can be the mean field theory still work, but when it can cause this emergence of a second eigenvalue outlier, then this framework breaks down. I missed the last point because the last point is still ongoing with Mei Yi here. I hope we can have that part out sooner or later. Thanks. Yeah. Thanks for the question. So yeah, so as this orientation-dependent connectivity, as Li Chun said, there are some special version of the spatial dependence. Okay, I cannot say our model can capture all these things. What I can say is, if technically, if we want to modulate this specific type of heterogeneity, depending either on the orientation selectivity or on the spatial, we can like treat them as separate neuron population. Like if you have this pinwheel structure, we can approximate this pinwheel structure as, for example, 8 different populations correspond to a specific orientation regime. Like each, they are different with each other. Yeah. Yeah, then...
Then my question would be, when you sort of do the mean field, how many actual neurons, right, you can consider this would be, right?
Yeah, good question. So the mean field, I think Professor Huang should know better than me. The mean field has the existence of mean field or accuracy of the mean field depends on how large the n is.
Yeah.
Right? I don't know. It should be infinite large. Then the mean field is accurate. Numerically, yeah. Yeah, 100 or 1,000. And regarding Xingyu's question, that's a limitation of this kind of framework, I think. It's because we always assume homogeneous with some specific, we call them population, either functional or connected. Yeah, it's... And also, I think Stefano used to mention to me that because even the data from Alien Institute that connectome data set, they're missing like just 2,000 neurons recorded. A large part of the connectivity matrix is missed. Right? Yeah. Yeah. You know that work from Yuhu. No, so that the decomposition implemented by Professor Yuhu is one branch. So it's one method you can use. Another, I think there is another field like they mostly focus on or interested in this cycling, and they use another type of decomposition or mathematical things. And I read a paper by a previous postdoc with Eric Gabriel Oker, and actually he used this Feynman map and diagrammatic framework to do this. It's like you have this quantum, like kick one node and then some information propagate, and he used the Feynman map to describe the propagation and the information. I didn't know Feynman map. I just know the name, yeah. But if you are interested, you can find the work. Functional. So functional, as I said, the low-rank framework is a functional. And actually, if you think about kappa, the kappa mentioned in my first talk and also Bing's talk, that you can treat kappa as a computational unit, and which you can also correspond it to a functional unit, functional population. But it's not like this work want to address, because I want to bridge the biological connectome data set to this low-rank thing. And yeah, you remind me one thing. So I didn't answer the question about whether the correlation in the motif and the correlation in the low-rank thing, because surgeon kept receiving the question like whether you can find this correlation in the real data or in the real biological things. This might be an answer. Like the local correlation in this very tiny or just one pair of synapses can be linked to each other through this, for example, chain way or reciprocal way, and then contribute to the generation of the sigma NM. That is the global correlation. Yeah.
So the Tau C you set it up at 0.1. So
yeah.
How large is that?
I didn't give the equation. So it is a Pearson correlation, right. Actually. A Pearson, so divided by the standard deviation. So it depends on here. So actually why, so here I use tau C is 0.1, and in the previous figure I used tau R reciprocal as 0.1. I have this analytical expression for this, and the tau C should be in the scale of square n. In that way, the network can be stabilized in this weakly connected regime.
Okay.
Yeah. And for the reciprocal, there is no square n. So that's why the effect of reciprocals is very small compared to the chain. And also, if you consider like z squared, the reciprocal is just a diagonal line, right? Yeah. But the chain motif can be off diagonal line. The abundance is not in the same order. So that's why they are. You should have. Thanks.
Okay, thank you.
All right, welcome back. So in this lecture, we're going to discuss the application of the Hebbian and Anti-Hebbia that we discussed on Tuesday through two examples. One is that we want to generalize the framework we discussed to supervised learning tasks with multiple layers of neural networks. The other example, we'll use the Hebbian Anti-Hebbia model as a toy model or simple model to understand a phenomenon called representation drift. So just to recap what we discussed on Tuesday. So we started from some, which we call normative approach, right? We postulate the object function, which we call non-negative similarity matching. So this is what it looks like, right? X here is your firing rate of the input, is a high dimensional vector, and you want to generate some output, vector y, such that the inner product of your pairwise input is closely matched by their corresponding output vectors, right? And we add some constraint on the firing rates of the neurons. So this objective function, by using some mathematical tricks, you can transform this original one to a new object function where you can use gradient descent and ascent to derive both the neurodynamics and the learning rule for the synaptic weight. And then you can map that variable onto a neural network where you can interpret the matrix variable that introduced as synaptic weight. And then it gave you this very simple neurodynamics, right? And a very simple learning rule, which is basically a Hebbian-based or correlation-based learning rule. So we briefly mentioned on Tuesday that this kind of network can perform several different types of unsupervised learning, such as sparse spatial learning, manifold learning, and non-negative independent component analysis. So this is what we left on Tuesday. So today I'm going to show two examples where we can extend this framework to either address more challenging computation tasks or to understand some experimental phenomena. So in the first part, we are going to address the supervised learning by using this framework to design a biological plausible learning rule. I think it's obvious and important that the question is how does the brain solve the computation, right? What's the algorithm and how does the brain solve computation task? Because although we know that the major form underlying learning involved selective plasticity, but the question is any learning process will involve a huge number of neurons in the synapse. So how does the brain control individual synapses to achieve some global object? So this is also called the creative assignment problem. And this problem is solved in machine learning by this backpropagation arrow algorithm. As most of you have heard about that. So for example, if you have multi-layer feed-forward network and you train to do some classification tasks like image classification, the way you update the synaptic weight is by calculating the cost function, the derivative of the cost function with respect to the synaptic weight. For example, if I want to update this synaptic weight, right, you can present some input, you generate some output, you compare with your desired output. That gives you error signal, which is E, and you take the derivative of E with respect to W, and you can get a closed form mathematical term describing how you should change W. And this involves two terms. One is the activation of this node. The other one, which you call back-propagate error at this node. But if you look at the error signal here, right, on this synaptic weight, it depends on all the error of the next layer and the synaptic weight of the next layer. So in that sense, you have to implicitly have information of all the other synapses and neural activities. So this is an non-local learning rule. And the other property of backpropagation is that there is two separate paths. One is we call a forward path, where you present some input, you generate some output. The other one is backward path, where you calculate the error signal and backpropagate to the previous layers. And the other constraint that there is a requirement of the synaptic weight between your forward path and your backward path. So all these constraints make it very hard to think about how could the brain implement such kind of algorithm, right? But we do know that brain can learn very efficiently. So over the last few years in the field, there's an explosion of work trying to address like whether we can release this kind of constraint to design learning algorithm to approximate backpropagation. And there's a great progress has been made in the field. So a few years ago, when I was interested in this problem, I was thinking about, okay, maybe an alternative approach is to think about slightly different object function instead of directly approximate this kind of algorithm. So that is came to how can we generalize similarity matching framework to supervised learning. The algorithm that we had in mind was motivated by the observation also in the brain and also in deep neural network trained for image classification. So this is the figure I showed on Tuesday, where if you look at how human monkeys represent different objects, right? Along the ventral visual pathway, if I present different like human face, body parts, different animals, fruits, daily items, and the V1 level, you look at the representation similarity, you don't see clear structure here. But progressively along the hierarchy, you can see in the inferior temporal cortex, there is a clear sort of category information already there. For example, there are two big blocks where this part corresponds to animate objects like human face, body parts, etc., and different animals, where this part corresponds to inanimate objects. So in that sense, if I were to decode whether an object is animate or inanimate, it's basically binary classification, I can easily just read out from the neural representations, right? So this is also the case if you look at a deep neural network trained for image classification and at different layers, you will also see this progressively change of representations. So from long structure representation similarity to more categorical informations. So this motivated us to think about, okay, maybe we can come up with some intermediate learning objective, right? For example, if I want to design some learning algorithm to do image classification, the goal is to gradually morph the representation similarity at the input level, which you don't see clear structure from the pixel level, right? And you also know the desired output. For example, in this handwritten digit recognition, at the end, you want to classify whether one digit belongs to one category. So the desired output is kind of block structure, right? And if you can successfully learn some intermediate representation, that can extrapolate between the previous layer and the later layer, and you should be able to success to achieve this goal. right? This is kind of the motivation we had. So how do we do this? And we designed an object function that contains layer-wise similarity matching. For example, the input is pixel value, and the desired output may be some one-hot encoding of different category, 0001, the only one element is non-zero. So the first part of your object function contains the pairwise, the layer-wise similarity matching, as we just discussed, right? You want the representation similarity at this layer close to the previous layer. But this is a supervised learning, so you have to contain the learning signal. So how do we add this learning signal to the object function? The easiest way you can think about is just adding some mean square error on the last layer. So here, let's say this YT is a representation of your desired output. If I see a character 4, right, it should record produce 0001000, or this kind of one-hot encoding, right? And RP is like the output model. And the last term is not essential, it's just a regularizer to make the algorithm to be more robust. But mm-hmm. You mean this one? Or?
Right, I mean, don't we want the distance between two different vectors at layer n-1 be similar to the distance between two different layers? Should we have a t at a t-1 or something?
Oh, yeah, good point. So, and it's actually equivalent. So, so you're saying maybe I need an object function such that this one matches the average between this one and this one, right? Something like that.
I mean, here you have the dot product between the same vector, so it's just the norm of the vector. So you're just saying that the norms of the vector.
Ah, okay. Sorry. Good point. So the indices here, okay, this should be T prime. Yeah. Yeah. So the pairwise similarity. Sorry about that.
T and T prime.
Right. So it's any pair of input. Yeah, good catch. Sure. Thank you.
And then you put them.
So in order to derive some log question. The last term, the regularization, you can have different regularization here. For example, you can have L2 or L1 norm of that. So that will give you slightly different activation function. Let's say you want to generate some ReLU activation function, then your norm of the last term should be L1 norm. If you have L2 norm of that, you will have slightly different object function, but so slightly different activation for each unit. So the idea is that you cannot satisfy both this term and this term. That's the point, right? So for example, if you want the output layer to be exactly at this one, then you cannot have a representation that is close to the pixel level similarity. So this is kind of a competition between two forces that enable you gradually morph the representation toward the desired block-like structure. So that's basically the idea here. So in order to derive an online learning, there's a missing piece here. Because the Hebbian rule or Anti-Hebbia rule was designed just to perform unsupervised learning. So we brought some very old idea in computer neuroscience. This is a trick, one of the methods that you can train an arbitrary network, either a feed-forward or recurrent network. This is called contrastive Hebbian learning. So the idea is that you can operate the network in two different phases. One which we call a free phase. So you present some input to the network, right? Let's say there's a group of input nodes. And you run the neural dynamics that's describing how each node activity evolve over time. Once it converges to a fixed point, you record them, which we call, let's say, each node x, I star. And then there's another phase called clamp phase, where you fix output to be the desired output you want. Let's say in the image application, I want output maybe 100, right? This is my desired output, and my input is the pixel, maybe handwritten 1. And then the network will converge to a slightly different fixed point, which we denote as x hat. So with these two information, the free phase fixed point and the clamp phase fixed point, then you can use the very simple Hebbian-based learning rule to achieve the task. So geometrically, people have been demonstrated that this is a process more or less like it gradually shapes energy landscape. For example, we are operating two phase, right? And initially the desired configuration in the state space, x-axis, is not a fixed point. Because otherwise you already output what you want, right? So at the beginning, the free phase, the fixed point is different from your desired fixed point. So by using this learning rule, this energy landscape will gradually change. So you will eliminate the fixed point at the beginning, while creating a new fixed point at your desired phase. So that's basically the picture underlying this learning process. And there's a light paper by Shanshan in 2003 where they proved mathematically that under a very small learning rate, this learning algorithm is proportional or is equivalent to backpropagation. So it's a kind of mathematical guarantee under certain conditions it will converge to backpropagation learning algorithm. So this is the idea that we also want to borrow here. So with both the Hebbian layer-wise similarity matching with this contrast learning rule, so eventually we have this new learning rule which we call contrast similarity matching algorithm. So the notation here is a little bit heavy, but just remind you that this is a multi-layer network with effective recurrent inhibition, where the inhibition at each layer, LP, is updated through this anti-Hebbian rule, where the feed-forward matrix of each layer is updated through this contrasting Hebbian learning rule, because here the substrate beta means there is some clamp fault there, and 0 means a free phase. So here B would be the bias of individual neurons, right? Combine this both anti-Hebbian rule and the contrasting Hebbian learning. So this network give you some interesting feature where after training, each unit will have, so they have very sparse representations. And then we can test this whether this work for like real machine learning tasks, right? The first one, the simplest case, we can test this on MNIST handwritten digit data set. So these are just a demo where when you have one hidden layer or 3 hidden layers. So the red line is this contrast similarity matching algorithm. This column is training error, and then you also have validation error. So overall, I would say it behaves similar to the existing variation of contrast Hebbian learning, such as equilibrium propagation algorithm. So the other thing that is very natural, you can generalize this learning algorithm to a structured connectivity, right? For example, like in convolutional neural network where each neuron may only receive input from a part of the neurons from the previous layer, right? So in this case, it's also worked very well for this MNIST data set. So this is kind of one piece of application that you can generalize the idea about having anti-Hebbian network to do some supervised learning. And this network have interesting feature where I didn't show here. It's basically the representation is more sparse than the case when you train a artificial neural network to do the same task. And the algorithm is related to the energy-based learning algorithm. But there's one big caveat here. that is, this algorithm doesn't scale very well. So for example, if you use this algorithm to train more challenging tasks like ImageNet, which is like a data set have more than 1 million images fall into 1,000 categories, then it doesn't perform very well. I think this is a general feature of all the current existing biological plausible learning rules compared with backpropagation. We are still very far away to get close to the backpropagation algorithm. Yeah.
Can you just remind us quickly about how biologically plausible the CSM rule is? I forget which layers need to be combined and which times. What are all the signals that have to come together for this learning rule?
Okay. So the recurrent collection, right? So let's say you look at the synaptic here, it only requires the pairwise neural activity, which is presynaptic and postsynaptic neural activity, right? And then you have decay term. The tricky part is actually the contrastive Hebbian learning. Basically, you have to store the information of the free phase. So you have to operate in two different phases. So one possibility people have been proposed that there may be some large-scale oscillation where you can think about, maybe at the bottom of the modulation you are doing free phase, or at the top of the modulation you are doing the clamp phase. The other proposal is that maybe it's related to sleep or wake cycle. So in a wake phase, you are doing like basically more like a clamp phase, where in the sleep it's more like free phase. So but sleep or wake cycle is very slow. So that's the very challenging part, how you interpret this contrastive learning in a mobile logic.
That would require some very long-lasting eligibility.
Exactly. Yes. So I don't think there is direct evidence that you can do this. This is more like in principle, it's local, it's operate online. So this is kind of property of the algorithm. Yeah. Other questions? Yeah. Yeah, good point. So the question is, can this network after training extract hierarchical features from image like in our ventral visual pathway? We know that the primary visual cortex, you care about the edge, the orientation, then you have part of the object, and gradually build up more complex representation, right? We only test this in CIFAR 10, which is 32 by 32 pixels. And we don't see clear hierarchical features there. As I said, because if it works for ImageNet, then it's easy to address the question you ask, because ImageNet has like larger dimensional input, so you can think about there are more like lower level features to higher level features that you can track. But unfortunately, the network algorithm doesn't work very well, so we didn't observe that hierarchical representations in this network. Yeah. All right, so now I'm going to switch gear to provide another example where this Hebbian and Anti-Hebbia network provides very handy tools to address some of the recent observation experiments, which is a phenomenon called representational drift. So the terminology of neural representation has appeared again and again in some school. By now, I think you are very familiar with it. But to be honest, I don't think there is a unified definition what exactly neural representation means, right? So for the purpose of this talk, we will interpret it as the neural activity patterns that encode external stimuli or internal states. And those activity patterns have causal relation with either perception, behavior, and memory. So specifically, if you think about single neuron, you can interpret it as the tuning of neuron to external variables. Like in the primary visual cortex, the simple cells they may tune to the orientations of different sensory stimuli, right? Or as a population, you train an animal to perform certain tasks. When we see this population represents something, that usually means that you can read out the task-relevant variable from the population activity. So that's, in that sense, we call the neural representations, right? So a natural question you can ask, are stable memories and stereotype behavior always associated with stable neural representations? So for example, we know the long-term memory or some motor task after learn can last lifelong, right? You learn how to ride the bicycle and then you don't need to practice. You can basically know how to ride it. So in the last 10 years or so, due to the technical advance, we can record a large population of neuron in free behaving animals for a very long time, maybe over weeks or even months. And a lot of the longitudinal recording shows that even in the constant environment, constant behavior, you still observe a continual reconfiguration of neural representations. So this is the phenomenon termed representation drift. And then the natural needs to a lot of questions, right? So you can ask, how does this kind of phenomenon emerge? What's the biological implication of this? Is like just some inevitable like biological bug, or there is some biological function, right? So we set out to address this by focusing on the mechanism under the phenomenon. But before that, we'll just give you an example, which is an early example, show this kind of phenomenon. This is a study more than 10 years ago where you put an animal in a constant environment, it's a linear track, over multiple days. So the animal become already very familiar with its environment. So let's say maybe every day at 9 o'clock, all the other control conditions are the exact same. You record the C1 region play cells for like 1.5 months. These are recording sessions indicated by the red bars here. So you already know play cells that we mentioned multiple times. Basically, if the animals are in this linear track, there will be some neurons that are active, right? For example, maybe on day 5, you can identify around 200 neurons. They are active, which means whenever the animal is at a certain location, there's some cells that are active, right? It's called place field. And you can order them by their receptor field. So in that way, you can kind of cover the linear track by these neural activities. So which means you can easily read out, as an ideal observer, you can read out the animal's locations, right? But if you stare at the same group of neurons in the following days, like 5 days later, the same group of neurons, same order, you can see some of them disappeared. Others may just shift their tuning locations. And this continues to evolve over time, right? But what's interesting here is that on any given day, you can still identify maybe slightly different subgroup of neurons, also around like 200. So this group of neurons may be slightly different between this one. And they have the similar kind of a tuning. So in this sense, we see that the place cell ensemble is very high, is dynamic, and the place field drift over time. And this has been reported by many other groups using different recording techniques and setup, even in virtual realities. And similar phenomena since then have been reported in many other brain regions, like the posterior parietal cortex during sensory motor tasks, and some, the odor representation in the peripheral cortex, and image recognition in the visual cortex, and the sound representation in auditory cortex, etc. So it seems to be this is a pretty generic phenomenon, right? So then it naturally leads to the questions about how this is emerge, right? What's the underlying dynamics of the drifts? Are there any biological implications? There's all tons of questions you can ask. So we set out to address this question by asking what could be the simplest reason or cause to produce this kind of representative drift. A few years ago, we had a hypothesis which was motivated by the observation in the brain that the synapse is very volatile in the brain. So even under constant condition, without explicit learning, there is ongoing turnover synapse has been reported by many experiments. So we had a hypothesis basically says, like if you think about neural circuit that is involved to some learning task, or we call representation learning, and if the synaptic dynamic, there is ongoing turnover of synapse, or you can just see it as some synaptic noise, and if there is some degeneracy of your object function, then it seems to be inevitable to observe this kind of representation drift. So this is a hypothesis, right? So you are looking at a certain group of neurons, maybe here, and this is responsible for some representation task like physical location or some task variable, etc. And record them over days or even weeks, right? You can ask why their representation evolved even though the readout or the behavior is stable. So geometrically, you can think about the hypothesis like this, right? So in a more theoretical way, the learning process can separate into two phases. The first one is a relatively fast time scale, right? You start with some initial configuration of your synaptic activity, and then you quickly find a solution, maybe here, which we call configuration space. So here, the two horizontal axes represent the parameter in your network. Usually, this could be the synaptic weight between two units, right? And the vertical axis is your performance, right? You want to lower your performance as some object function. But if the object function has some low, sort of degenerate manifold, that gives you, roughly speaking, the equal performance, right? Like this valley along this energy landscape, right? Any turnover or fluctuation of synaptic weight will drive the network to explore this equivalent, which we call degenerate space. And in experiment, you are not measuring this. What you are measuring is neural activity, right? So you can transform this into the neural activity space. So this manifold will have a corresponding manifold in the neural activity space, which is the shaded region here. And this gives you a continuous evolving neural representations. So this geometric picture says there has to be some confined subspace where the drifting activity is reside in that space, right? So that's basically our hypothesis. So to test this hypothesis and make predictable predictions, we were focused on brain region where neurons have, we call localized receptor field. Face cell is one of the examples, right? There are many other examples you already heard about from Albert talk, like the head direction cell system, where neurons are tuned to the heading orientation, right? Even the bird, like the owl's auditory pathway, I think the midbrain, where you can identify neurons that are tuned by the parameter defined by the inter-oral time difference and level difference, basically how the sound enters the left and the right ear and the difference of intensity. So they can form this auditory map, which is usually used to locate the prey. And you also heard from Xin's talk, like neurons in hippocampus, they may tune to more complex behavior relevant variable, like the task evidence and position conjugate to the two variables, right? So in all these examples, there's a unified property of this receptor field. Basically, there's bump-like tuning curve, and they will tile some underlying manifold. This could be some low-dimensional variables. And then it's easy to study representation drift because the drift could be quantified by looking at how individual receptor field moving around, right? So that's what we're going to focus. And we want a model that is as simple as possible, that can capture the essence of a hypothesis. Remember, we're talking about some representational learning where the underlying solution space has some degeneracy, and there is ongoing noise, synaptic noise there. And again, we want model to be interpretable in the sense that all the turnover synapse should be a biological plausible process. So there's naturally need to the Hebbian anti-Hebbian network we discussed. So I'm just flashing out the network that you're already very familiar with. Just pointing out that previously there's been mathematical proof that depending on your data manifold and the number of neurons, after learning, you will see that each output neuron only responsive to the input that comes from certain region of your input data manifold. So that's the idea. And here, we are interested in the fact that in the long run, if there is some ongoing, either you call turnover or synapse there, synaptic noise there, how would the individual receptor field evolve over time? So that's what we're going to do. So the first example, we are going to use this to model hippocampal place cell formation. So in rodents, right, we know that the hippocampal place cell, especially those in the C1 region, they receive input from the entorhinal cortex through two different pathways. One is called direct pathway, the other one we call trisynaptic pathway. And the entorhinal cortex has neurons that exhibiting this characteristic hexagonal firing pattern, which is called grid cells. And different grid cells are organized in a way such that you can have different modules. Each module is characterized by different space separation, like which we call scales, the orientations, and also the offset to the origin, right? And I don't think there is like definitive mechanism underlying how exactly place cell forms, but there has been an early proposal that one possibility how the C1 region formed the place field is like this, right? The pyramidal neuron in the C1 region, they receive direct input from a high-dimensional sensory input that is encoded by discrete cells. And also, those pyramidal neurons, they compete with each other. So this anatomy closely resembled the Hebbian anti-Hebbia network that we just studied, right? This is basically a single neural network with some input and with lateral inhibition. So that is naturally we thought, okay, maybe we can just try out this Hebbian anti-Hebbia network to see if it will give us some meaningful results here. So how do we model place cell formation using our network? So given environment, as we said, either agent or an animal there, you can have a high-dimensional grid cell representation. I'm standing here. There is some firing pattern in my entorhinal cortex encoded by grid cells. I move around. There are other neurons that encode this position information, right? So the input to our model is a high-dimensional representation of the physical location there. These are just illustration about different modules based out, right? This larger space short scaling, this smaller scale here. Now, you can imagine there is some agent roaming around in the pyramid. Each time you receive a vector encoded by this grid cells, right? And you update your synaptic weight over time. So what's going to happen if this network converges to your configuration, right? And after that, you can look at the reset field of output neurons, right? So not surprisingly, you will find for those that are active, they have this characteristic single bond plate field like receptor field. Again, there are some neurons are silent, right? And if you locate the centroid of them, you put them together in the original arena, right? You can see that they cover the space quite uniformly. So that's the picture where the simple Hebbian anti-Hebbian learning could produce a place map over a given environment. Right? Good point. So in the object function, in fact, if I go back to the original, So you can introduce slight modification here. So instead of match the exact inner product, you can add some, a constant here. Let's see. That basically means you only care about the representation that is close enough. Let's see, there may be some threshold of 0.5. That means because it's non-negative, right? If the inner product is smaller than 0.5, the best strategy is just output 0. So in that sense, you can control the tuning curve, the width of your receptor field. If that's narrow enough, then there's only one possible active bump. Could be the solution. All right, this is all good. We have learned the receptor field from gray cell representations, right? The next step you can ask, okay, what if I introduce the synaptic noise to our online learning scheme, right? Previously, in the previous slides, so after learning, this will be stabilized there. Each will pick up a location to represent the physical space. But now, there is ongoing synaptic noise in both the forward and the recurrent synaptic weight. So what's going on? So next, I'm going to show a video where you can track 10 output neurons to see how it evolves over time. So that's the typical phenomenon you observe both in the simulation and it's also similar to those found in experiments. So you can see some of them are relatively stable at number 10, right? But the others are more transient. They are active for a while, can move around and disappear, emerge again, maybe at slightly different locations. So this is kind of qualitative feature about the behavior using this network as a model for beta cells. So to make a direct comparison with experiments, which most of them are done in linear track environments. So we modified the model a little bit. The trick here is that, the gray cell representation of linear track now is not periodical. It's actually a slicing through this hexagonal 2D patterns. Right? So anyway, you can do that. You can train the network. Not surprisingly, after training, those that active will cover the linear track. For example, if you mark the location from 0 to L, these are the maybe 200 active cells. Each of them have a bump receptor field will cover the linear track, right? And again, because of the noise, this repetition will drift over time. But you can ask how similar as a population my output neuron will represent location 1 and 2, right? Because individually you will see there's no constant repetition for 1, no constant repetition for 2, right? But you can ask what the pairwise relationship, where it change, how it change, right? So the simplest case, you just define pairwise similarity by their inner product. This is what you found, right? Although the representation population activity drifts over time, but their internal pairwise structure is constant, right? It doesn't evolve over time. And qualitatively, you can see what happens in the experimental data, right? This is the population activity across about 2 weeks. This is the pairwise similarity, which is more stable, right? So the other thing to quantify and compare with experiments, like you can look at the representation each time. Let's say whenever the animals are in this exact location but different time point, right? You can ask how this vector compared with the previous time point. Right? So because it's drifting over time, so it's different every time the animal at these locations. But you can quantify by defining a population vector correlation coefficient, right? So it's kind of an exponential decay to some low value. So this is what we found in experiments. And the other features, which I mentioned at the very beginning about the original experiment, that is, there is a constant fraction of cells that are active at a given time. For example, both in the model and in the experiment over 3 weeks, roughly speaking, there is a constant of cells that are active, right? Because the beta cell can disappear, can re-emerge again, maybe at different locations. This, and can we make any testable prediction, right? So one prediction we had is by looking at the drifting correlation direction. So you can ask, if there are two active phase cells at this location on B1 and that location, I want to predict in the next stage where you observe they move on the same direction or the opposite direction, right? This is, because in linear track, this is the only possibility, right? So the model predict depending on the separation distance. So if they are very far away on the linear track, the next day they are more likely to move on the opposite direction. So that is like quantitative. You can have the red line here depending on the separation or distance, right? But as a neural model, you can think about if they just move around independently, then you don't see this distance-dependent effect, which is the green line here. So we analyze very carefully about data sets here. And so in this experiment, you can see these are the active phase cells over like 2 weeks, right? This is the normalized location of linear track. And no, on the zero, there are relatively fewer of them active, but then there's a constant fraction of them active. So if you track one phase cells over 2 weeks, this is basically the trajectory, right? So it moves around quite a lot, right? And you can compare maybe this one, right? See, day one, they are here, day two, one of them here, one of them here, right? And day three, one of them here, the other one there, right? So based on all this pairwise information, you can compute a line like the red line here, and this gives you the experiment result. I think it agrees very well with the model prediction here. So the other prediction we have is that depending on the tuning strength, right? Because some phase cells have larger phase field, others have more stronger tuning but lateral, etc., right? And we found in the model that for those that on average are more stable, it turns out that, for those that are more strongly tuned, actually more stable over time. So the metric that we use in this case, you can define a one-dimensional diffusion constant, right? For example, Brownian motion on an axis. So the diffusion constant, larger one, means on average will drift faster. So there's a clear negative correlation between the mean amplitude of the receptor field versus the drift speed. So when I was preparing this slide, I noticed one of the very recent study where they used the virtual reality setup. And this is a very interesting paper because they tried out very different conditions, right? They add a lot of different sensory cues trying to address whether the observed rapid drift is due to some hidden unobserved variable that unrelated to the spatial location. Turns out on any contingency test, you always observe rapid drift. But anyway, so in their experiment, they separated two groups of neurons. One they called stable cells, the other one unstable cells. So this figure shows that receptor field on the day where it starts to emerge receptor field, right? This each row is a different trials. And they found that on average, those stable over long run tend to have larger receptor field. So here, these are the 3 consecutive days, right? The day 1, day 2, and day 3, etc. Those that have lateral tuning turns out to be very unstable. And this is just like 2 different ways you can define the tuning strength, either by just looking at the calcium signal strength or the integration over the curve, right? So statistically, you can see those stable cells have stronger tuning, right? This is qualitatively very similar to this prediction, right? When I'm still analyzing to give more like quantification about whether we can get something like this figure. maybe it's a good time to pause for a while. Any questions for this experiment or model prediction? All right, so this model can also explain other brain regions like the posterior parietal cortex, where you train the animal to perform a simple teammates task based on the context the animal experienced first half the stem, right? For example, if the animal see a white wall and black dots, it should turn left, otherwise you turn right. So after training, this animal can perform this task very stably over several months. And then you record the neuron in the posterior parietal cortex, and there are neurons, group neurons are tuned. Basically, they're active whenever the animal is a certain location of the neural tract. And the model can capture all these phenomena and the statistics about how many of them are active over time and how the fraction of active over time. So quantitatively, this provides a possible mechanism to explain the drift in this brain area. So I'm going to summarize the second part, where we propose that the representational drift could be attributed to a learning process with degeneracy and synaptic noise. With these two ingredients, it's inevitable to produce representational drift. And we test this using the Hebbian anti-Hebbia for both hippocampal base cells and the posterior parietal cortex, and make several tests of prediction. Some of them are consistent with later experiments. So I want to point out that the framework we used, the similarity matching or Hebbian anti-Hebbia, seem to be very general. For example, this is also a recent study where they look at how the auditory cortex neuron respond to different kinds of sounds, pure tune or more complex sounds, over a time course of 2 weeks, right? Again, first you observe this kind of drift. These are just the population tuning curve. But the pairwise similarity over 2 weeks, almost the same. So looks like the brain trying to preserve certain pairwise similarity structure here. One comment I want to add is that the model that we propose is just one of the possibilities. For example, there are other models that can be explained what happened in hippocampus, and it's consistent with some experiments. For example, there are studies showing that if you want to store memories in the hippocampus continually, the inevitable effect that after storing new information, then the representative of the old information changes. So that's one possibility. The other model is that you can build a model with slow fluctuation of intrinsic stability. So that gives another fixed mechanism why the repetition changes over time. So I still have time. Maybe we can discuss a little bit related questions, like because all the experiments we reported or discussed has the feature that either the memory or the behavior is constant, and all the experiment conditions that you can control are the exact same. There are natural questions: how do you reconcile this constant behavioral memory versus this very volatile representations, right? So with the model that I showed here, we explored the degeneracy idea. There is some underlying submanifold that is degenerate. But you can imagine there are other mechanisms that you can withstand this change in representation. A straightforward one you can think about, maybe the variation is confined in the direction which people call the null space. For example, in this very cartoon figure here, if you want to read out the summation of the firing to y1 and y2, so any variation along this line doesn't matter because you always get the same readout, right? This is called coding null space hypothesis. So the other hypothesis is related to the population code, which means what matters maybe is the geometric structure of population representations. For example, in the teammates task, maybe what you want to present is the task structure, which is whether to turn left and right, and also the locations, right? Where you should turn. So you can maybe withstand some rotation of your population pattern, but you don't have to be the exact same. But it doesn't address how exactly you can read out from this rotated representation, right? So there's still another question here.
Yeah, so what do you mean by stable behavior here? Because so from the theoretical point of view, it's very clear that if you have place field code and there is a grid, then you can decode very well. You can train a decoder and you can learn very well the location based on the activity. So what is it?
Okay.
I mean, my question is, do we know that actually the behavior of this animal is stable?
Yes.
It doesn't depend on there's no time history effect. So how careful we have been in the data analysis to claim that the behavior is stable? Because the performance can be stable, but the behavior encompasses way more things than the performance, right? The scratching.
Right. Yeah. Agree.
Many other things.
Yeah. So this is a very good question, right? So you can never sure that there is with the experiment today and tomorrow that you control everything, including not only the physical environment but also the internal state, the emotion of the animal, whatever, right? So I think that's very hard to address. But what I can see is that from different labs with different setups. So for example, one of the most important factor that may affect the sort of the arousal state is basically body movement actually have very strong influence on all the other brain regions, right? So even you exclude that kind of contributions, you can still find this kind of consistent drift. The other thing related to what you said, like how can you achieve stable decoding? So at least in the hippocampal base cell scenario, you cannot have a fixed decoder. If you as ideal observer, you can train a decoder, but only on data from every day. So for example, if you train the decoder from data on day 1, it doesn't perform very well 2 weeks later. So you have to retrain again. So in that sense, that's what I mean, either the brain has to come up with some idea that you can track the change to continue to update that. Or at least from a static decoding point of view is very puzzled. So that's the point. Oh yeah, okay. Yeah, maybe it's not a good idea to say stable behavior because you can not for sure that other part of the behavior is stable. So what I generally mean that is the task performance or the memory that people thought that this because there's a causal relation because you can initiate the population of cells, then the performance drops drastically. So in that sense, we call maybe a stable performance would be a better word. Yeah. Max?
I want to follow up on that because in the 2 experiments you highlighted, the Schnitzer lab linear track experiment and the Carlos Boyd's lab linear track experiment. The animals didn't have to do anything. They just had to run back and forth on that.
Yeah. So no. Yeah. So in that sense, forced task, right? It's more like a free behaving, just behave like itself. Yeah.
So we've been collaborating with Carlos Boyd on reporting in the labyrinth. And there the animals do have to do something. They have to remember where stuff is and to recognize it the next time. And we find that the place fields are much more stable over 3 months back.
Wow. Yeah. So does that mean?
I think it's partly because they are locked to the environment and the animal really has to work with the structure of the environment in order to accomplish something. And so, following up, I love your treatment of this, but I wonder, can one add somehow a feature to the cost function that breaks the redundancy, basically? That adds a cost for deviating from a stable feature of the environment or something like that.
Yeah. So basically, you want to add some anchoring point in your speed space. So one possibility, you train animal to perform multiple tasks. Because you can imagine if you train only one task, there's a huge subspeed that can perform that task. But if there's a demanding of other tasks, then you actually gradually shrink the possible solution, right? That's the idea for machine learning. Then one of the predictions that if you train to perform multiple tasks, then you will observe slower drifting rate. That's one of the possibilities there, yeah.
Another perspective we can look at here is the example that we saw that
Mhm. Yeah.
That little bit of variability that is representation actually adds strength right to the tuber stability. You can train a network for it much less pressure than if you rely on X-ray.
Yeah. Yes. All right. So okay, maybe this last slide I can add is, I think this is also important questions like, are there biological function or what's the implication of the drip, right? So I think in the field there's a lot of debate there. I just will flash out some of the ideas. So one thing that people have been arguing that this kind of representative drip provide a mechanism doing Bayesian sampling. A couple of people talk about Bayesian inference, right? Similarly, so if you want to train a network to perform certain tasks, you don't want your configuration to stack to one particular solution space, but rather you want the network to continually explore that space. So what's the benefit of having this behavior? One is that in principle you can do posterior calculation, which is in general is more robust, like to noise. The other thing is like if you perturb the network, let's say if I kill some of the units, with this mechanism, the network will rebound very quickly to achieve a high performance. So that's one idea. So the other idea, we just mentioned that maybe this is a side effect for some continuous learning, right? Because we're never sure that during the separation time, the animal is doing something else, right? So, and there are recent studies showing that by incorporating the drip mechanism, it's doing something like implicit regularization. It's a term in machine learning where regularization also enable network to perform more robust. So the other idea, which I am actually exploring, that this can provide a mechanism maybe to integrate different memories in time. For example, that memory happens today, maybe two weeks ago, right? They are separate in time, but maybe they are related, right? So how does the brain eventually integrate these two memories, right? What kind of mechanism, biophysical mechanism to enable that to do that? So in the serial memory engram, so this trait provides the possibility that because you have two representation of two memories, if they are continually evolved, then there's a chance that they have large enough overlap, then you can integrate them. So these are just like several ideas in the field. I don't think any of them have been materialized, so just keep in mind. So and last, I would like to, my collaborators, Genghis, Leontar at Harvard, also David, Shiva, Arneva, and the Mitya from Flatiron. Thank you. Yeah. Yeah. We actually added all of them, but we actually also explored like if you only have noise here and here, so the effect is actually slight difference. I don't remember what exactly. One of them have larger effect compared to the other one. For example, if you only add noise here versus if you only add noise here, is one of them has weaker effect compared to the other one. But I don't remember which one. Yeah, sorry? No, I can check the paper, but we have discussion there, so yeah. I'm not sure I understand. So you are seeing what kind of feature can we learn from this model about T cells? About stable cells. Oh, okay. And there are actually experiment evidence showing that. So if a neuron has higher intrinsic excitability. That's actually a topic, a hot topic right now in current, I would say. So let's say all the neuron has some baseline intrinsic excitability, let's resting potential, right? And everyone is fluctuating over time. But by any chance that you have a higher membrane potential, then you are more likely to become a place cell and stabilize there. So that's one possibility. But that's not in our model. So our model only can predict that if you are more stable, which means like on average you have a stronger phase field or stronger spatial tuning. That's what we can say from the model point of view.
You also about the shape. So I think some people wonder if there's a species difference. And rat people thought the rat value are more stable, because less is than light. I don't know what to take about this.
Yeah, I'm not very familiar with the experiment data in rats, but I'm sure that in the flying bats that people have been found, and 3D place cells seem to be quite stable. So in that sense, there is indeed some difference between different species. But I would love to hear from the rats like experiment if you know some of that. Yeah.
Okay, let's have a 10 minutes break, then we go to the life science discussion session. During the 10 minutes, you can still ask questions.
All right. Thank you.